Okay, good morning, everyone. I want to welcome you all to the 14th CNAG Symposium. This year we picked Cancer Genomics. As you know, those of you who've been at these things before, we sort of pick a topic that is specific for things that we do at CNAG, but then we bring collaborators, people we work with in to give presentations about work that we've done with them. Today we have a really exciting program moving forward, but importantly, this event happens with the sponsorship of many different companies. So we have Illumina, Oxford Nanopore Technologies, Agilent, Bonsai Lab, and Roche that are supporting us, and we appreciate this very much. It really makes the event possible. In addition, I have to acknowledge funding from our funding agencies, which is the Spanish and the Catalan governments. And I'll say that we also have a lot of funding from the European Commission, and that we are part of Omics Tech, which is a research infrastructure, Spanish technological research infrastructure. Okay, program today is very exciting. I will just flash it through quickly. A couple of talks before a coffee break, a couple of more talks before a lunch break, and a couple of more talks after that. You have to stay to the end. Because at the end, we will give a little tour of the facilities for those of you who want, so you can go and have a look at sequencing instruments. Unfortunately, today you will not be able to go and look at the CPT, where we have the computers. The reason for this is we have an upgrade going on. Even the sequencers, we're having upgrades going on. So you'll probably see sequencing instruments in boxes as well, if you go to that. But we have the CPD is full of boxes at the moment because they are unpacking new computers that we bought. Okay, yeah, as I said, it's the 14th symposium, and just a quick run through CNAG. So CNAG was funded 15 years ago, so you've noticed we've done one less symposium than years we are old. However, the mission that we have has not particularly changed over time. So our mission is to do research projects together with the research community that lead to improvement in people's health by one or another means. So this can be as much trying to make headway on cancer, trying to make headway on rare diseases, trying to make headway on immune-mediated diseases, or trying to produce nicer oranges that you then can put in your orange juice and that are good. So we do a lot of things across the center. We came here 15 years ago with one mandate. We had to support the Spanish contribution to the International Cancer Genome Consortium, which was on chronic lymphocytic leukemia. That project, has been a huge success. I think Spain, in terms of genome research and cancer genomics, really put itself on the map with that project, because it was a project that, out of all of the ICGC projects, it was the first project to publish a finding that was issue of that particular large project. But it also was the first project to complete, having done 500 tumor genomes of people suffering from chronic lymphocytic leukemia. So we've made a lot of headway on chronic lymphocytic leukemia, and I won't go into details. Elias Campo has given several talks at these symposia and explaining where we've gotten in the meantime. I'll say a little bit something right at the end of the symposium. Okay, what is CNAG? CNAG is 135 people. We represent 22 nationalities. We publish or contribute to about 125 research articles every year, and we have something between 60 and 70 competitive grants. So we have money that comes in through that, and this is essentially PIs who are asking for funding and then carrying out projects. The areas that we work in nowadays, as I mentioned, we started out with a mission to sort of look at chronic lymphocytic leukemia and cancer and so on, but we opened that up into other topics. So we brought on board in about 2012 rare diseases, have built a very nice system to handle rare disease genomics. We've transitioned that informational know-how to the impact projects that are the Spanish projects with the objective to sort of install genomics into a personalized medicine system. Then I think also by 2013 or something like that, we started doing single cell analysis very, very early. I think Holger joined us in 2015, and then since then single cell has been growing an awful lot. And then on top of it, we've now got Ana Pascual, who's sort of taking care of the single cell work when you stick it down on a slide, and then you figure out how cells are communicating with each other and what they're doing. She will explain that a lot. Immune mediated genome in action is just a different word for epigenetics, if you want. So that is all about having one genome sequence, but multiple different functions in relation to having conformation of the DNA molecules organized in a certain way, or having methylation that sort of tells you what is going on or what is active inside these cells. As I said, immune-mediated diseases, that's turning into a really big thing. Obviously, there's a lot of overlap with what you do in single cell and what you would do in cancer when you look at immune-mediated diseases. There is that overlap. And then biodiversity is a topic that we picked up about 4 years ago through a project called Biogenome Europe. And for that, and through a Catalan project called the Catalan Biogenome Project, we are sequencing species that are of interest. These are ones that are important for our environment and so on. Okay. How are we organized? We have two big units. The sequencing unit with 4 different teams underneath it, and the bioinformatics unit with also 4 different teams. Sergi heads one himself. And then we have a quality system that runs around this activity. Sequencing capacity, this is always difficult to figure out how much we actually have. It's about 20,000 gigabases per day that we can do, and that equates to about 200 genomes that we can do every day. This is actually ramping up for 2026. We're entering into a project called the Genome of Europe, and we will be doing an awful lot of genome sequencing in 2026 for that particular project, probably nearly at a rate of 200 genomes per day. At the bottom, you can see the different applications that we use with our sequencing instruments. So basically handle genomics, transcriptomics, epigenomics, single cell, and spatial. Quality is a really important factor. From the beginning, we have certified ourselves for ISO 9001. We got ISO 17025 about 2 years later. We got that, I think, in 2014 or something like that. And since then, we've been maintaining the 17025. Importantly, last year we achieved the ISO 27001, which is an ISO for data security. And it turns out that this is actually really essential because, for example, accessing data from NIH requires you now to have ISO 27001. This was not something that was 3 years ago, but now they are requiring that you have that, so that you have a data center that is secure, no one could break into it, and so on. So we do that. We have a couple of other ISOs that we're working on getting. One is 15189, which is a clinical ISO. This needs, for us, we need approval from the Ministry of from the Cat Salud. They need to approve us as a healthcare provider, and then we can move ahead and get the technical stuff in place so that we can get ourselves accredited according to that. Okay, we do many projects. So we run something like 1,100 projects every year. A project can be small or a project can be quite big. So it is an awful lot of things to handle, and the teams that are handling this, if you've done a project with us, you know that you usually start talking to project management person, and then afterwards you sort of move over to maybe getting one of the specialists drawn into a conversation, and once you know exactly what is being done, then you end up with the samples that are transferred in a certain way to us, handled by us in a certain way, quality controlled, prepared for sequencing, taken through the sequencing instruments, taken through data quality control, and then handed to you. Or if you want us to help with the analysis, we help with the analysis as well. So out of the 135 people we have about 70 people who are computational, and depending on what the project is that you're trying to do, you can get support from someone who is specialized in the type of analysis that you would like. Sequencing instruments. This has been moving in the last couple of days. So we have currently I think 6 Illumina sequencers, 2 NovaSeq X Plus. I think one arrived yesterday or today, I don't know. And we have still 2 NovaSeq 6000s. We have 2 MySeqs. We have the Pacific Biosciences sequencer. This is a Revio, and then we have, I think at the moment, 4 Oxford Nanopore technology sequencers. I think 2 of them are arriving tomorrow or the day after. So we have 2 new coming in there. So a lot of production going also onto nanopore sequencers. In the single cell space, we have 10x Genomics Chromium Connect technology, and also the 10x Genomics Chromium X. Spatial, we've got quite a few machines on that. We have, I think, 4. These are sort of high-resolution microscopes with a lot of fluidics hanging off it. We have 2 Bruker NanoString Cosmics instruments, 1 10x Genomics Xenium, and we have a Bruker Vutara. All of those we know very well how to use. And there's a few new things coming. We won't tell you about that, but we'll tell you later once we have them. Interesting stuff. Okay, compute capacity. We have 2 different types of processors, CPUs and GPUs. GPUs you all know because you all use ChatGPT and and so on, artificial intelligence. That's the kind of processor that these large language models are built on. The CPUs has been reasonably constant. However, there is always an upgrade. So we always have to sort of we always have to maintain them. Processors tend to become inefficient over time because older generations and so on. So we replace them with new ones, and the same goes for the storage. So this is all at the hands of our analytics teams. Then, in terms of the research, there's 8 of us, teams and groups. Mark Marty, who's here. Tyler, I haven't seen. Simon Heath, Holger Hein is here as well. Anna Esteve, I've seen as well. Anna Pasquale, you will hear afterwards. And Thomas Marquez, he's not here, but he's going to get a big prize tomorrow in Madrid. So we're looking forward to him getting that. Really delighted about that. Okay, so with that, I will sort of kick it off, and it's my great pleasure to introduce the first speaker, who is Sabine Tetschba, and I just hand it over. You know better what you talk about than I do. So.
That's mine. Yeah. Thank you, Ivo. Hello to everyone, and thanks, Sabine, because she's already sort of introduced a bit what I will also be showing you, a bit from another angle perspective. Just again to mention that was a collaborative effort that we've undertaken in the last couple of years together with Holger, Sabine, and Bruker Spatial, the R&D team, who basically allowed us to use non-commercial assays and allowed us to sort of help them develop what we can now do with it. Also in terms of analysis, I'll be talking about that later. So the aim was to basically take whole tissue sections, curated, manually selected by Sabine and Tim, to try to understand the molecular, cellular mechanisms and the architectural traits that change together with colorectal malignancy. And so it's not only important to have these big tissue sections in which we have this reference or healthy mucosa, pre-malignant lesions, and tumor lesions. That's what Sabine and team selected carefully. But I think that the intestinal epithelia is also a perfect model system to study a lot of these things, also because in health, you have this transcriptional trajectory that basically aligns with the physical axis. So you have the stem cells in the basal side of the epithelial layer, and then the fully differentiated enterocytes in the luminal side. And this then allows us to have in one space, in one physical 2D axis, all of this information, all of these transcriptional identities all at once. And we want to understand exactly how that happens transcriptionally, morphologically, also bringing in always this histopathological, let's say, background and notion. And that's exactly where also Irene from the team, our histopathologist, took a great time to really carefully annotate all of the tissue sections that Sabine sent us, to have this very well annotated cell by cell in terms of these reference mucosa, pre-malignant lesions, tubulovilus adenomas, and the tumor lesions. In 8 different patients, 8 different tissue sections that we then analyzed further. So Irene also assessed and classified the different sections in terms of grading, differentiation characteristics, and staging, invasiveness. And with that, we also characterized or classified the different patients in this 2D scheme, being the upper right corner, let's say the most malignant cases. But then that's the histopathological characterization of the tissues that we were analyzing, and on top of that, we started running the whole transcriptome assay. And basically with that, what we get is a 4-protein panel that allows us to not only see, let's say, the morphological characteristics and the cells in the tissue, but this information will also then be used by machine learning-based algorithms to segment single cells from these tissues. And that's what you see here in the lower panel. So based on the protein stainings, we define single cells in these tissues. And since we have these almost 19,000 RNA targets analyzed from the same tissue sections, we can then assign each target to each individual cell. All in all, we had around 3.5 million cells, huge pieces of tissue, a lot of data. And what we did then, Ginebra from Holgers took adjacent tissue sections, quite thick tissue sections, to basically do single cell RNA sequencing that we then, Helena, used as reference profiles to basically annotate the cells in space. We used in situ type, which is an algorithm developed for this type of data, and we basically could resolve quite nice heterogeneity both in the epithelial compartment, in the immune compartment, and in the mesenchymal compartment. So we had a good representation of all cell types. But all in all, the first thing that Helena saw was that basically in the broad compartment level, epithelial, immune, and stromal, there was already a robust increase of epithelial components in the tumor lesions compared to the reference and the tubulovilus, while the immune compartment decreased. When we go a bit deeper and try to actually do compositional analysis in higher granularity, we see that in the epithelial fraction, there's these fetal-like populations and stem-like populations, Sabine has talked about them previously, that really appear already in the tubulovilus adenomas and accumulate then in the tumor lesions. In the immune compartment, we see this accumulation of tumor-associated macrophages characterized by SPP1, MRC1, and C1q. And in the mesenchymal compartment, we see a strong accumulation of cancer-associated fibroblasts, the yellow population that you see there. That's not the interesting one. The interesting one is the dark blue, which you can barely see. Those cells, the myocaph cells, are very low abundant in general at the tissue-wide scale. They account maximum for in between 1% and 5% of the mesenchymal population across patients. But they will become very important when we zoom in particular regions of the tissue that we understand are key in cancer progression. I will show you about that later, but these myocaphs are basically characterized by FAP expression, CTHRC1 expression, and they show a sort of contractile phenotype and matrix remodeling phenotype. Again, related to what Sabine was saying in the extracellular matrix compartment and tension forces mechanic stress. Okay, what was also interesting is that Helena saw that there was an association in between these either fetal-like or stem-like phenotypes with, let's say, grading. Helena also wanted to interrogate a bit the microenvironment, so the niches around each cell. So she took a 50 micron radial neighborhood around each cell and measured different sorts of things. One was density, again linking to this concept of forces, of compactness. And what we saw is that from reference to pre-malignant to malignant, there is an increase in epithelial density in particular. And that went together hand in hand by a decrease with malignancy in terms of histopathological assessment. There was a decrease in entropy, so linked to a more homogeneous epithelial composition in cancer lesions and a higher degree of homophilic interactions. And she also clustered these niches to then see a broader architectural view on cellular organization across sections and across histopathological regions. And while there was this invasive front that was clearly unique to tumor regions, this sort of onion-like layered architectural forming traits was actually quite consistent across tumor and pre-malignant regions. The same cell states or cell populations were actually being increasing from the core building niche towards the invasive front in terms of the fetal-like phenotypes, MMP7 positive, the CAF myofibroblast, and the tumor-associated macrophages. But then Helena said, I would really like to look into tissues, not only, let's say, comparing discrete regions in terms of niches or in terms of histopathological annotations, but I also want to look at the niche in a continuous way. And that's why she basically used now the fact that we have this ground truth in the tissue, in which we know that we have this trajectory very well defined from stem cells in the basal to fully differentiated enterocytes in the luminal site to basically test trajectory analysis in space. And that's what she did, and basically resolved what we knew already very well, and this sort of built up confidence on then interpreting further how the reference mucosa indeed goes into the tubulovillous adenomas and then to the tumor regions in red. So now we can also ask, okay, we can track this epithelial malignant transformation across space and now across time as well, and we can ask how the tumor microenvironment co-evolves with this epithelial transformation. And that's what you see in the three last bottom graphs there. So on the one hand, we have these stem-like and these fetal-like epithelial cell states appearing already in tubulobulous adenomas and then getting larger in the tumor lesions. We have tumor-associated macrophages also being most abundant in the tumor in the later pseudotimes, and that's the tumor lesions in space. And then the cancer-associated fibroblasts. Again, you barely see the blue myocafs appearing in the plot below. And now it's not only about cellular composition, it's also about a bit more going functional and trying to score different pathways, different modules along this pseudotime, and then also asking which exact receptor and ligand pairs are enriched along this trajectory, and how we can track these changes basically in cellular interactions. And I will not go in depth into all this, let's say, more discovery sort of plot. That's what we are now doing, trying to look deeper into that, using orthogonal methods to validate some of these interactions that we've sort of identified at the transcriptional level, but that's now ongoing work. I hope we'll be able to talk about that later next year. But what was interesting is, since we had this broad tissue view on different donors, Irene could basically spot that there were repetitive sort of units across these broad tissue sections that were sort of, let's say, strange. And they were strange because in one single crypt, in one single unit, you could basically see two very distinct morphologies. And what Helena did is to try to see if these distinct morphologies, one cell next to the other, were also related to different transcriptional identities. And that's exactly what we have there. So that's an example of 4 of them, but to show that in a single crypt, we have reference mucosa, one cell, the next cell is already either tubulobulous adenoma or directly a tumor cell. And that's morphologically so, but that's also transcriptionally so. And also interestingly, the microenvironment around these different 2 poles of a single crypt also changes. That's a bit also what Sabine was showing in terms of these abrupt transitions that might come from fusion events or fission events. We obviously don't know, we just have a snapshot of it. But they are quite abundant in the reference mucosa, in the premalignant lesions, and interestingly also within the tumors regions. Also, how the pseudotime nicely marked these different transitions or malignant transformations. And then next step after transformation is potentially invasion and metastasis. And that's actually what we also captured in this study. So we basically had lymphovascular structures in which we could observe clusters of epithelial cells located within the lumen of these vessels. So Irene did a massive work also annotating each of these single events. And then Helena sort of transcriptionally monitored those events, both from the luminal side, so which epithelial cell states do we have within these lymphovascular invasions, but also around those, then compared to the more broader tissue view. And what we did is to compare these lymphovascular invasions also with invasion-free lymphovascular structures that were found, let's say, very close to the invasions. And that's the visual representation of it, but I think it's more clear if we look at the bar plots, because you'll see that basically we have quite predominant identity of these fetal-like MMP7 populations forming these invasions. Around these invasions, we have an enrichment with tumor-associated macrophages, but what's most striking is that dark blue population that was barely seen in any of the tissue-wide plots that I've shown you so far accounts for almost 80%, 85% of the mesenchymal population around these lymphovascular invasions, suggesting a key role of these fibroblasts in particular mediating these events, which are crucial to metastatic spread. And from one of the patients in which we had lymphovascular invasions detected by Irene, we basically had also a lymph node that Sabine has also shortly shown, so I'll go shortly through that. But so there's a lymph node from a primary. This lymph node has two very distinct growth patterns. Growth patterns that we also see in the primary side. So we have quite well differentiated mucinous type of growth in the bottom right corner, and we have a more undifferentiated neuroendocrine pattern in the left. And this we can relate to the primary of the same patient. And transcriptionally, when we just analyze, let's say, the tumor fraction of this lymph node and forget about the rest, we see clearly also behaving very differently transcriptionally. So we have the right side of the metastatic lymph node being stem-like in terms of transcriptional profile, and the left side being fetal-like, mapping also the different identities that we resolved in the primary site. And the question is obviously open, what drives these differences, and can we see that in other patients? So that's what we are now also working on. But now to finish, just to emphasize that Helena didn't only do a great job analyzing data that no one else know how to analyze, basically, but also she took a lot of effort and time on making this data publicly available for anyone who wants to use it, check, or develop tools upon that. I think it's a super rich resource, so please go check it out. And I just want to thank everyone involved, Sabine and team, obviously Holger, Irene and Helena, who basically did the work, impressive team, and Ginebra for all her work on the flex and the single cell part. The Bruker Spatial also for allowing us to use their assay. And just a teaser, I would love you to have you all next year in the third conference for the European Society of Spatial Biology that will be co-organizing together with Xavi, Miguel, and Edu in Sitges. And with that, I'll finish, and I'll just take your questions if there are any.
Thank you, Anna. Questions?
Gracias, Anna. Have you guys studied very well what happens in these sharp transitions that you have in the tissue? Because that's surprising to me.
It's so I have to say there's already some reports talking about these abrupt transitions, let's say from a morphological or histopathological perspective. They've been, and maybe Edu can then comment on that because it's not my field of expertise at all, but so they have been related to normal, let's say, homeostatic mechanisms of the healthy colon mucosa. They've been also related to a proliferative burst in premalignant lesions, but so far there was not much known about these transitions. We call them that way, right? I mean, they might just be fusion or fission events. So let's say two crypts that are fused into one single daughter event. And what we are basically capturing is the identity of the two mothers, in a way. Or the other way around. Yes, Edu.
Yeah.
So I think one of the problems is, I mean, we see tissues in 2D.
Absolutely.
Right, but what we know is you get these tumors that also start invading the healthy crypts.
Yeah.
And then when you section, what you see is many times is the interface between the tumor and the healthy. But not necessarily this interface implies that the healthy goes into tumor, right? It's simply that the tissue start invading and then it eats the healthy, the tumor eats the healthy, and then you actually capture those interfaces, but not necessarily a transition from normal to tumor.
But Edu, in this case, we were very careful. I mean, not me, obviously, but there was a lot of care. There was a lot of care put on really taking the basal lamina as ground truth for being absolutely sure that indeed what you were capturing is one epithelial cell next to the other and not having a 3D artifact on that. So, and in the transition crypts, it's exactly the same. So you basically see the basal lamina. So it's...
Yeah, but this is well studied. You see this, well, we can talk later.
Yeah, absolutely.
These tumors, there is all this bottom up, top up, pre-morphogenesis, right? And then you see, that they get, we see this in mice too. And then you have this impression of a very sharp boundary, but does not reflect transformation from one to the other.
So I totally agree with your point, and that's exactly why we are really pushing to go now in three-dimensional.
I think that's probably very interesting.
Absolutely.
Thanks to you. So.
Thank you for the talk. It was very interesting.
Thank you.
I have two questions regarding the annotation. You mentioned you had to develop a reference from a normal tissue, or how do you...
No, so what we did basically is from the same blocks, we took one section to do the spatial assay, and the next section, we smashed it, Gene did that, to do then single nucleus RNA sequencing from it. So we would have the best match single cell reference that we could to then be able to, the best we could represent these classical single cell readouts in terms of clustering annotation back in the same patients in the tissue then. And we used in situ type for that, which is an algorithm that allows also to use, let's say, either fully unsupervised or fully supervised, semi-supervised type of annotation from a reference.
Okay, thank you. And the other was more from a statistical point of view. I suppose the challenging of all this is how many areas or how many patients or tissues do you need to represent the biology, no?
Absolutely. So probably you've seen that there's no asterisk in any of the graphs that I've shown, basically because we have 8 patients so far. Sabine has a lot more of them, and we are indeed enlarging the cohort, and we are basically validating also what we've observed in a lot more patients. So in terms of statistics, you are completely right, but you still can make, let's say, discover potential mechanisms that you can then validate in statistical meaningful cohorts. That's a bit what we've done, or what we are trying to do.
Yeah, thank you. Very interesting.
Okay. Thanks so much, Anna. Okay, so we move to a presentation by Andres Ramirez. I've seen him. There he is. So Andres is from 10x Genomics, and he's going to tell us about the machines that they give us. We buy from them.
Sorry about that. Good morning, everyone. I'm very happy to be here again to talk a little bit about the tools and the technologies which fuel all these amazing discoveries. And it's going to be a little bit of a recapitulation of what happened in the last year and a half, roughly speaking. And I have to start defending why we charge you money for it. It takes a lot of effort, hard work, and of course resources to become a leader on this field. It's a highly competitive, high-tech environment that's very fast-paced from the perspective, the evolution of such tools. So in a short time, so much new technology has come into your hands, so you can come up with your amazing discoveries. And I want to briefly acknowledge this, explain to you that a lot of this comes from our efforts, remembering we started very humbly on a garage, into becoming one of the world leaders for single-cell and spatial omics. It did not have that accent. There's a lot of work on the innovation side. We have a very strong R&D team, of course, who are the minds behind all these technologies, their developments and upgrades. But there's also a very strong customer support, which is something that characterizes 10x. And I think that's also something that kind of sets us apart from the reality in the market. We have very good scientists working hand by hand with you on the field in your labs, helping you make the better use of these technologies. Because tools are just tools. You need a mind that can know how to use those tools to get good results and good data. So talking a little bit about the tools, 10x has built basically an ecosystem of different tools. And this is where everything started with Chromium, which is our single cell franchise, let's say. And Chromium, it's a very well established and has quickly become, not so quickly really, it's kind of like a bad adjective, but it has become, we're very proud to say that we have become the gold standard for single cell transcriptomics. And many reasons for that, which start by the wide variety of applications we can offer. From the multi-omics perspective, not only you can measure gene expression, but also you can detect different proteins, either on the cell surface or in the intracellular compartment. CRISPR guides perturbations driven by those CRISPR interventions, TCR, BCR, clonotyping, and more. But we are also compatible with different sample types. We offer protocols for handling and preparing samples, starting from tissue, and then through different types of preservations. We can be species agnostics if you are into that, if you want to drift away a little bit from traditional mouse and human models and work with something more exotic. Different types of sample types. You can start with cells, you can start with tissues, fixed, non-fixed. So there's a lot of freedom and space for innovation from your side to work with our tools to make a very good science. So this has been the foundation for years. However, technology is taking us in a different direction now. And as we go deeper into clinical, the problem becomes scale. Because it's not easy, but let's say it's more manageable to work with a few hundred, a few thousand cells. But as you start coming up with larger studies and different patient cohorts and such, the scale becomes an issue. And scale also means that we need to be able to manage the cost, right? And this is something we have invested time and effort, and we have come with a few innovations which are worth mentioning. First, the ability to do on-chip multiplexing, which is something that's already available for either our 3' and 5' chemistries. Now, you can use the benefits of our newer chemistry, GEMX, to have increased sensitivity on the gene detection, better throughput for your cells. And what's interesting is now you can multiplex up to 4 different samples on the same chip. You collect them collectively on the same well. And this eliminates the need for upstream tagging for your samples, so it makes your workflow a little bit easier, more straight to the point. And you can work with the different sample types, and it's also supportive of lower sample inputs, so you can work with different sizes of different sample sizes. So this was interesting, but we're still under pressure from the community to try to come up with even larger scale studies. And in order to address that, we have taken our flex assays, which are well known for being very compatible for preserved fixed samples, which is very important in order to schedule your experiments properly, acknowledging the need of being able to deliver a product that's compatible with typical clinical workflows. You don't know when you're collecting the sample sometimes. It might come from a biopsy today, tomorrow. So FlexV2 gives you the ability to now multiplex on a plate. It's interesting from the sample handling perspective, but also this unlocks new avenues because plates are more friendly and compatible with automation. So if you are into larger scale studies, this is definitely something to keep an eye on. We are all for flexibility, so we try to be more flexible not only with the number of samples, but also the number of cells, the sample types. But we aim to offer you the same multi-omic readouts that you have been able to obtain through our Flex technologies. So a few numbers here. So number of cells has been scaled up, so now it's possible to analyze up to 1 million cells per channel. Working with plates gives you the ability to work with between 2 to 384 samples, so we aim to offer you the ability to work up to more or less 1 million cell screen per week, 384 samples. And what's important, and it's interesting also to mention, we try to reduce the waste, so you can reuse your samples. It's not like you start an experiment, you use just half of a plate and you need to discard everything. You can reuse those unused plates and restart the workflow when it's necessary. So you can really use this technology to scale your work as your samples are coming, and we try to keep it flexible as we can. So some developments trying to scale up studies, trying to bring single cell to larger studies, following what's happening in the field nowadays with projects like 1 billion cells. And now going into our second area, which is spatial. Just a few words about Xenium. Xenium has also come a long way since its launch. We started also with a platform we were aiming to be also among the best, and we are convinced that we have become among the best. We are known in the field. We have been benchmarked by researchers, so we are not participating on this, but we have been described to be more sensitive and specific than other platforms available in the market, showing strong correlations with single RNA sequencing experiments when you compare orthogonally. And we offer a platform that's incredibly low noise, so you can really go deep into your cell characterization with single cell resolution on your tissue without compromise. We aim to be pretty flexible from the panel perspective. So we offer different choices of customization. We have a wide variety of pre-designed panels. All those genes have been curated to try to answer questions which are strongly focused on oncology, which is the topic of this symposium. So for either mouse or human models, we have tissue-specific or tissue-focused panels, which you can very easily customize, adding up to 100 extra targets on top of those. And then we have these more exploratory panels with 5,000 genes, which can also be further customized with up to 100 genes on top. But not only that, it's not only about the repertoire of genes you're trying to explore. You might have more specific questions. You might be interested in different forms of transcripts. We also offer that. We offer you the ability to detect differentially on your tissue, map spatially different isoforms which are expressed or could be expressed in your tissue. If you want to work with xenografts, that's also something we can offer. Exogenous sequences, either viral sequences, if you want to overexpress reporter genes on your tissue, it's also doable. TCR, PCR clone typing, very interesting for addressing questions about the microenvironment. And single nucleotide variants, also fully compatible from sample perspective. There's a lot of variety to choose from, whether you work with fresh tissue, paraffin embedded tissue sections, cell suspensions, something very interestingly shown in the last years. Also CNAG very actively involved with this stamp procedure as well, and organoids. So a lot of variety and just a snapshot of what's been done in last year. So we have a wide variety of applications. Starting on the upper left, we have exogenous sequences. Some of them are in the upper panel. We have the cervical cancer, where we have been shown it's possible to detect genes which are expressed after infection by HPV, so detecting oncogenes which are originally encoded by the HPV particle in tissue blocks from human. something we hope is not going to make a comeback soon. COVID, also by viral sequences, it's possible to detect in lung tissue organoids. And then if we move into splicing, we have a detection of different isoforms of the receptor 8 for EGF in glioblastoma, single nucleotide variants as well, and then some exotic non-human models, tilapia brain, and plant biology. So a lot of flexibility, a lot of compatibility to make these tools usable. We also did some work not only from the compatibility perspective, but also to be able to segment your cells better. So we brought a few years ago multimodal segmentation, where we offer you improved segmentation. We started with nuclear expansion, now we do multimodal. And what's newer, which was launched this year, was offering you the ability to go deeper into true multi-omics with the ability to co-detect protein on the same tissue section. So you can run on the same platform, on a single platform, you can detect both RNA and proteins as part of the same experimental run. This is available at the moment for fresh human tissue. You can detect up to 27 different protein markers. Customization, of course, is on the roadmap, is something very important. We are actively working into it, in the same extending the panels into mouse. How does it work? It works very well. So in here, just an example on renal carcinoma. As you can see, there are different insights that can be offered by protein, which can be overlaid on top of your RNA readouts for purposes of validation sometimes, but also for purposes of guiding your hypotheses into deeper discovery. Sometimes in this case, it's a good example of where RNA-based clustering might seem to be giving you a ground truth in terms of these two regions seeming to be quite homogeneous. However, adding proteomic readouts give you a better insight into potential differences from the phenotypic level, in this case explained by a difference in the expression of CD138, which is a marker typically associated with a bad prognosis in different tumor types. Secondly, clustering can be done either by RNA or by protein. This is done by Genome Explorer automatically. As you can see, you can cluster your cell populations by protein expression, by RNA. And faithful to our core principle, the technology does not require pre-optimization from your side. It's been tested and validated on a wide variety of different human tissues, as you can see, ranging from intestine to liver, breast, lymph nodes, glandular, non-glandular tissue. So it's been tested to work in all tissues. So we are very excited about this product, and we are hoping to see what discoveries this brings to the table. Moving into Visium, which we could say is the older brother of the spatial CNAG family. Visium also, it's been longer out there in the market, and I would say probably you are all familiar with Visium HD, which is our way to approach unbiased discovery from a spatial perspective. So it's a sequencing-based technology that's whole transcriptome, and we work to increase the spatial resolution of the assay. We are going subcellular now with binning of data down to 2 by 2 micron bins, which give you a very good resolution, spatial resolution of tissue structures. Importantly, it's been validated to work with FFP tissue, fresh frozen tissue, fixed tissue, different types of tissue. It's also been pre-optimized. It works quite well. However, cell segmentation is always an issue, a topic with sequencing-based methodologies, and we have worked this year to bring you into the native software the ability to do cell segmentation based on nuclear expansion for Visium HD. You just need to have your H&E images from the same tissue section, and then the software is able to track the position of those nuclei there, and then reconstruct the cell morphology or approximate the cell morphology by nuclear expansion. What for? Because it allows you to reconstruct better your structure, the morphology of your tissue, which is very important to answer those special questions. A good example of that here, as you can see, resolves better no matter the preservation type. Can be FFP, fresh tissue, fixed frozen tissue, but what's really interesting is to give you better insights. As you can see here, this is a tonsil sample. As you can see, there seem to be two main clusters in the interstitial space. However, when you look at the methoxylin, eosine, you can be suspicious that there is actually more. And when you do nuclear expansion, you can resolve much better those cellular entities that are present here in the interstitial space of this sample. So overall, a great addition to the software. That's, of course, free to use, free to download, and it's available for all of you. And then another pending story with Visium, because Visium HD is probe-based, therefore it's a little bit more rigid. It's available for mouse and human. You can also spike in your own custom probes, but not so flexible as we would like to. And we worked this year to bring the foundational chemistry for Visium, which was the Visium 3'. Maybe you remember these large 50-micron spots that were the manual placement of that, so it was a little bit more hands-on. But now we're bringing Visium 3' to HD, to high definition, allowing you to do whole transcriptome in a species-agnostic manner. So this opens all sorts of new avenues for research. Not only you can now maybe study the spatial distribution of, let's say, isoforms of different transcripts, but also you can go species agnostics. You can work with xenografts, models, and so on. It's also been tested and validated on different tissue types. This is available for fresh frozen tissue as it's poly-A capture, but we have tested in different non-human models. As you can see, primates, we have worked with canines, rats, chicken, zebrafish. So for all of you working maybe in development, maybe working with xenografts, it's also a very interesting tool to consider when your questions become spatial in nature. Of course, tested as well in human and mouse, but we all know about that. And a good example of how it works, this is from a work where some human cells have been injected into a mouse model. And as you can see, we're able to resolve quite well genes coming from mouse and genes coming from human. On the mouse tissue section here, you can see how very well spatially how the human, in this case, cancer cells injected on the mouse model take over and form a tumor, and you can discriminate very well the cells by their origin, and you can study processes like, in this case, colonization of a tissue by cancer cells and resolve it spatially in an unbiased manner. So that's what I wanted to share with you. Thanks again for the invitation, and looking forward to see you next year.
Time for one very short question, and it's from me. When you scale in terms of the number of cells in the chromium, what price do we end up with?
Well, I don't want to be political, but as this is a question for our distributor, really, in Bonsai Lab. But okay. I can avoid the question.
Okay, sorry. Accept. Good. Let's move on. The next presentation is by Edouard Baillet. We've collaborated with Edouard for a long time. He didn't have to travel very far to come here because he's in an institute next door to us. Okay.
My presentation is in the...
okay.
Because
you know how to do it.
I hope so. Yep.
Perfect. Great.
Thank you.
Thanks.
Thank you. So let me see if that works, because I cannot see it moving. Well, anyway, let me start. Well, yeah, but it's not moving here. Well, anyway, so congratulations for the 15th anniversary, and thank you very much for the very kind invitation. I apologize, but right. So Yeah, very good. So, yes, Ivo is said, we've been having long-lasting collaborations with people in the CNAG. I'm super happy to be here and share some of the stuff we've done together over the years. And so my lab works on colorectal cancer. I just want to make the point that in colorectal cancer, we know a lot about the primary cancers, but this is, I would say, from a very pragmatical point of view, not a major clinical problem because most of the patients will get these tumors surgically removed and they don't die of the primary disease. So the issue is, of course, metastasis. Metastasis happen in about 30 to 40% of the patients, happens in vital organs, the liver and the lungs. And the problem is we don't have good medicine. So there's been very few approvals for colorectal cancer, new drug approvals, and over the past 34 years. And still the backbone is based largely on chemotherapy. And so, over the years, we've been building models in mice that reproduce of the human disease. And this is probably our last more sophisticated models where we implant organoids that carry driver mutations in the sort of the key oncogenes or tumor suppressors, APC, KRAS, P53, and the TGF-beta receptor 2. So we implant them in the cecum. They generate focal colorectal cancers that invade, and then we operate before metastases are present, and then we can track the evolution of the disease and the emergence of metastasis in these hosts. So what we learned, we've learned many things, but I think one of the most surprising things is that when we look at the sort of the very early lesions in the liver and in the lungs, just after resection, so that the residual cells that are beginning to metastasize, what we found is that these early lesions are heavily T-cell infiltrated. You see here the liver, and these are two small tumor glands, and the staining for CD3. So you can see really lots of T-cells attracted to these early lesions. But then as metastases progress over time and they become bigger, T-cells are actually excluded, and you can see them here sitting around. And so I'm just going to summarize our initial findings. So what we discovered is that these metastasis-initiating cells are initially recognized by the adaptive immune system, and the reason is because there is pre-existing immunity against the primary tumors in this cancer. So the adaptive system is trained to recognize the cancer cells. The cells get out of the primary, of the protective environment, and they immediately recognized by the adaptive system. We think these cells hijack, we have some data suggesting that these cells hijack interferon signaling, so that all these inflammatory responses are very high initially. They upregulate PD-L1, so PD-L1 is an interferon target, and they protect themselves initially from the attack of the adaptive system. So we can actually eradicate these cells initially just by giving checkpoint blockade in the neoadjuvant setting, so something that has been now tested by Miriam Chalabi and a few others. Right, and then over time, these cells are the ones that make it, are very good at attracting tumor microenvironment. And in particular, they attract CAVs and macrophages, and CAVs and macrophages are actually machines of producing this hormone called TGF-beta. And TGF-beta impacts on the different cell types in the tumor microenvironment and impose a gene program that we know it predicts tremendously well poor prognosis in colorectal cancer, something we discovered back almost when I started my laboratory was the initial finding. And then, so TGF-beta itself is some of the genes induced by TGF-beta in these other cell types then will block the action of the adaptive system and will enable the expansion of metastasis and the formation of full-blown mets in the liver and the lungs that look like immune excluded, as I mentioned before. Now, we had this beautiful collaboration with Holger and with Paula and the Holger team. And this is a project led by Ana Henriquez. In my laboratory, I sort of couldn't explain this in 20 minutes because this paper, which is published 2-3 weeks ago, it came up, right? Has so many models and how much single cell, which I decided is, I'm just going to give you the highlights in one slide, okay? So this is the sort of, but as I said, this is go and look at the paper because it's a wonderful, really wonderful collaboration. So this is the cancer immunity cycle. As you know, so tumor cells release tumor antigens that carry by dendritic cells into the lymph nodes, and there the immunity is trained. And so then T cells that are trained to recognize cancer antigens will migrate into the tumor, and then hopefully there will recognize the tumor antigens and eliminate the cancer cells. So this is the sort of the well-known sort of standard immunology in the field. So what we learned from this collaboration is that there are two sources of TGF-beta that impede this cycle. The first is a source in the lymph nodes that signals directly on the T cells, and that blocks the aggression of a subset of memory cells from the lymph nodes into the blood. So these are actually these cells that will, these memory-like cells that will migrate into the tumor microenvironment, and the cells that are proficient to eliminate the cancers, the cells that are sitting in the cancers are basically gone. We cannot resuscitate them with anything. So are the new cells arriving from the periphery, the ones that are really proficient. So TGF-beta blocks the aggression. And then there is a second source, Anna learned, a second source of TGF-beta that instructs a subset of macrophages to become immunosuppressive. And these macrophages now prevent the expansion of the newly arrived T cells into the tumor microenvironment. And we know that depends on the protein that is called osteopontin, a extracellular matrix protein that is induced by TGF-beta in these macrophages. And so the knockouts for the osteopontin, the knockouts mice creates metastases that are really T cell infiltrated, and we can really eliminate just giving a checkpoint blockade. Now, the important thing for therapy, as I said, this is a very, very short summary of probably 5 years or 6 years of work, right? It's just in one slide. So please, I mean, if you have interest, go here and read the paper and ask Anna and Holger, the details of the work. So the important thing is, so this process driven by TGF-beta is reversible to some extent. And so if we now take these full-blown meds and we give checkpoint blockades, essentially nothing happens. So I want to remind you, these are models of MSS colorectal cancers, and we know in patients checkpoint blockade does nothing in colorectal, MSS colorectal cancers. But if we now treat with a TGF-beta receptor 1 inhibitor, and typically we've used this one, galunisertib, then we see T cell infiltration and clonal expansion of T cells in the tumor microenvironment. And if we combine the two of them, there is a massive inflammatory reaction, and that results in the fact that the mice get cured of this disease. So control mice will die in a month, but the mice treated with the combinational therapy, just by treating 10 days with the combinational therapy, they actually will survive. They will die of age. So we had mice with 2 years that had metastases, they were completely cured and then survive. One of the questions we've been doing in the laboratory, maybe a little bit on the back, and is how we can push this into the clinics, right? Because there's such a robust effects on mouse, so we thought maybe the patients can benefit out of this. I mean, I don't have time to get into that, but we've been building companion diagnostics for TGF-beta, and also a number of different drugs that are now being pushed into the clinics. So one of the problems of the TGF-beta, I mean, all of the companies have had, or the big pharmas, TGF-beta programs. But one of the issues is that the very good TGF-beta inhibitors create massive bleeding, so massive cardiovascular defects, so aneurysms. And so the moment you systemically block TGF-beta robustly, the patients will die. I mean, the models will die because the good TGF-beta inhibitors never made it to the clinics. And so cardiovascular toxicities are always a no-go for companies, and all these programs have been blocked early on. And so something we did some time ago is we collaborated with Mirus. Mirus is a Dutch company just bought by another bigger company called Genome. So Mirus are probably the best in the world in producing bispecific antibodies. They're a really amazing platform to produce bispecific antibodies. And so what we did with them is design bispecifics that instead of blocking systemically TGF-beta signaling, like the small molecule inhibitors, they will block TGF-beta in particular cell types of the tumor microenvironment. That antibody, bispecific TGF-beta receptor to PD-1 that blocks TGF-beta signaling on PD-1 positive T cells was, but the program was purchased, acquired by Incyte, which is a big American pharma, and they pushed this antibody into the clinics, just released the data recently, a few weeks ago, And it's actually very, I would say, promising. So for the first time, we see that immunotherapy works on MSS colorectal cancers. We have 20% of responders, including many patients with liver metastasis. And so now I believe Insight will push this into a phase 3 trial, because that's kind of surprising for us. I mean, we only make this antibody, but it's super rewarding because that's sort of translation of 20 years of work in my laboratory on TGF-beta. Now, let me switch gears. So what about the tumor microenvironment? You've heard about this today, and I hope I can give you some more insights, because it's possible that it was a bit sort of confusing, right? So something we did a couple of 3 years ago is profile the tumor microenvironment of primary tumors and mets, both in mouse models and in patients. And indeed, what we found is these are tumors that are very heterogeneous, as Sabine and Anna pointed out. We identify a particular cell state that we call high relapse cells. And the reason we call high relapse cells is because these cells express a number of genes that predict poor prognosis in patients. So these cells are localized at the invasion fronts in tumor buds. We think they migrate collectively, enter the circulation probably as clusters, and then they seed the liver. So when we look in our models at the initial metastasis, they are made of this particular, of cells that adopt that particular cell state. This cell state we can label with a gene called EMP1. And then over time and through plasticity, they regenerate the other cell types present in the tumor. So in our models, if we genetically ablate, so we introduce a diphtheria toxin and ablate this population before surgery, then we save mice from metastasis. And that suggests or indicates that this is the cell of origin of metastasis in animals. Now, it turns out we were, and by the way, we treat with chemo. Now the cells undergo a bottleneck, and the resistant cells are also these sort of EMP1 cells that others have called them fetal-like. Right. So it turns out that, I mean, we are not the only ones discovering this. So different people sort of stumble almost at the same time into these sort of populations that they have received different names. You've heard a lot about the fetal cells. I have to say, very skeptical these are fetal cells at all. But this is a very fancy name. I recognize, fetal and even more oncofetal sounds like super cool. And we sort of keep telling these cells oncofetal. I think the evidence for any fetal state is very limited. Anyway, all these cells are labeled by this marker that we discovered, EMP1, and represents a malignant cell state that drives essentially what is clinically relevant, that is therapy resistance and metastasis in colorectal cancer. So there's a lot of hype onto trying to understand what the cells do, how we can eliminate them, right? So what we found is that if we look in the TGCA data, these are MSS colorectal cancers for the sort of the poor prognosis signature that these EMP1 cells express, and we ask sorry, I screwed up something here. Okay, if we ask, what mutations correlate with this particular signature, then what we found is that KRAS and BRAF mutations correlate very well with this. And if we now take healthy organoids from the healthy mucosa and using CRISPR, we introduce all the driver mutations, APC, KRAS. The only one that upregulates the sort of the poor prognosis signature is when we introduce KRAS mutation, suggesting that there is a relationship between the two. Now, we team up with this American company called Revolution Medicines. You may have heard of this. These guys are probably, I would say, I don't have stocks on them, so this is a disclosure. But I would say these are probably the winners of all the KRAS business. They have amazing inhibitors. These inhibitors are bipartite molecules. On one side, they sit on this chaperone called cyclophilin A, and then on the other side, they capture RAS in the ON conformation, and that particular one forms a cross-link with the G12D mutation of RAS, which is the most common mutation in colorectal cancer and in many other cancer types. So the company, Revolutions, have tested in PDX models for gastric, colorectal, non-small cell lung carcinoma, and PDAC, this molecule, and essentially what they found is very, very substantial results. Actually, that is being translated into patients, so it's beautiful data on PDAC. I would say for first time we see PDACs really shrinking with these inhibitors, very impressive. But it turns out if you look at colorectal, there's very mild responses here, right? So either cytostasis, so the tumor stops growing, or nothing at all. And they wanted to know what happens in our tumors. So, so we implanted our organoids in mice and then start treating with this RAS-on KRAS G12D inhibitor. And essentially what we saw is that we treat early on the metastases stop growing, but there's very little curative effect. If we treat later on, metastases keep expanding in the presence of the drug, very similar to what they saw in the PDX data, suggesting that these mets can survive without KRAS inhibitors. Now, what we discover is that when we profile these mets, there is a huge, there is not changes in growth, but a very prominent transcriptional remodeling. So what you have here is the vehicle, and in green, you have the cells treated with the inhibitor, and it was, very clear that these big metastases are basically made of these AMP1 cells, fetal if you want to, and then you treat with inhibitor, they don't die, but they switch into this other transcriptional state that is characterized by the expression of this marker gene called LGR5, and LGR5 is sort of the marker for cancer stem cells. So the cells transit from this poor prognosis state into a cancer stem cell state. This is maybe more clear here. So this is a EMP1 tomato, and LGR5 is seen by in situ hybridization. So you see EMP1 in the invasion fronts here in the Mets, and also sitting around the areas of necrosis. So we know there are these two populations of EMP1 cells. And so we treat with inhibitor, and now the EMP1 cells are gone, and we see the metastasis being repopulated by LGR5 cells. It's not one marker gene. It's a whole change in transcriptional program. So the whole stem cell signature is upregulated, and the whole sort of fetal or this poor prognosis signature is being downregulated. Now we can see these on organoids. That's actually quite cool. So these are organoids that we've introduce a tomato reporter into the EMP1 locus and EGFP into the LGR5 locus. So we can track the two populations. And now we treat with inhibitor, and you will see very rapidly the organoids move into these super LGR5 GFP states. We'll see if that works. Okay, so this is a movie with light here, but so you have the poor prognosis cells in red and the LGR5s are in green, and we treat with inhibitor. These organoids stop growing sometimes, but they basically become super LGR5. So what we're doing here is homogenizing the two populations into one single cell state. So we are basically getting rid of the heterogeneity in the tumors. Now, this is another collaboration with CNAG, and so that's been very, I would say, rewarding too, with Mark Marti and Mary Chale. And so Mark and Mary Chale has been looking at changes in the chromatin either over treatments or when we introduce different driver mutations. A huge data set I would say we don't understand quite well yet. Yeah, but the data, what we got here was very clear. So Mary Chale and Mark found out that the poor prognosis fetal EMP1 state is enriched when we do ataxic. In the organoids, it's enriched in FOS and YUN binding sites. Largely, this is this EAP1 transcription factor. And then when we treat with inhibitor, what we see is now that the genes being regulated are enriching these TCF binding sites. And remember, TCFs are the nuclear partners of beta-catenin in the nucleus. Now, Mark and Mary Chale have done high-C. I'm basically unable to explain this very well, but I think the bottom line here is that there is no, I would say, major chromatin changes or major topographical changes. So plasticity happens with minor modifications in the chromatin structure of the genes involved in this transition. This is just an example. That's beautiful Gaudi plots. I believe the cover, no, it's coming with this. Of the paper. So what we saw is, as I said, small topographical changes in the chromatin, but the AP1 sites around, this is EMP1, now will be closed. If we look at LGR5, again, little topographical changes, but what we have now is that the LGR5 locus and the regions around LGR5 locus in the genome, they upregulate or they open sites for these TCF binding sites. I'm just going to finish now. Now what we have is that colorectal cancers are made at least of two cell states, transcriptional cell states. So these cells have exactly the same mutations, but they can adopt these two different states. One state is dependent on KRAS and downstream the transcription factor AP1. This is associated with poor prognosis. We know these cells have kind of abandoned the intestinal-like program, and they acquire programs that are reminiscent of PDAC. And they can switch, when we block KRAS, to this LGR5-like state that is reminiscent to the healthy stem cells and is dependent on Wnt and TCF transcription factors. Now, my last just message is that we can, KRAS inhibitors do not cure, but we can use them to now play with plasticity. And we think this is important for patients. We can actually go and homogenize the tumors in a way that now become more responsive to therapy. And this is the last experiment. So here what we've done is we take an organoid and we introduce into the LGR5 locus, so that marks the cancer stem cells. We introduce a diphtheria toxin receptor. So remember, the mouse cells are resistant to diphtheria toxin, right? The human cells, all the cells are toxic, but the mouse cells are resistant. So now we made these LGR5 cells sensitive to diphtheria toxin and introduce a GFP. Right now, we use these organoids generate metastasis in mice, and we treat. So what we found is that if we treat with diphtheria toxin, we eliminate the GFP positive population, but we spare the LGR5 negative population. If we treat with the KRAS inhibitor, now we get rid of the LGR5 negative population because we push them into the LGR5 state. Now, if we combine the two, we basically eliminate the cells. And this is now shown in mets very nicely. So we treat either with the KRAS or with diphtheria toxin. We see a reduction in number of mets, but animals will sort of die. This is control. These are monotherapy. If we combine the two, now we basically eliminate a vast majority of the metastases. These animals survive long term. Some of these animals kept alive for a long time. So this is now our way of thinking is essentially all the therapies we have in the market target either one cell state or the other. And this is a major reason for therapy failure. So what we need is combinatorial therapies that are rational and treat the two states, or therapies where we can move the cells, treatments that we can cells that move in one direction or the other direction and then come with a second hit to eliminate. We're also trying now to push this into the clinics. Let me tell you, all the KRAS data is, for lots of people in the lab, but largely driven by Alessia Sentonze, with help of, as I said, lots of people in the lab, Adria, bioinformatician, and Xavi. And then the second part, very nice collaboration, led by Ana Henriquez in the laboratory with wonderful collaborators, the Marmarty Renome Lab and the Jorge Elhain Lab. And thank you very much, and maybe I've been over time, right? Sorry. No? Okay. Yeah.
Questions?
People need coffee.
I have
coffee
a question
now.
on when you use these combination therapies, are side effects worse because of combination therapies or not?
If you combine therapies, you may have worse effects. I have to say that these KRAS inhibitors are not toxic, or very, toxicity is very limited, which is surprising. So patients live well with them. We don't have yet a therapy for LGR5 cells, but we're looking into that.
So it's not in human yet at all?
Well, it's not in human, but it's not very far away from humans. So something we've done with mirrors, with this other company, we have another bispecific antibody, LGR5 EGFR. This is in phase 3 and approval trials for head and neck, and also nice results in colorectal. And so that's possibly a combinational therapy we're going to use.
Sounds very good. Questions?
I have just one. It's a Nice question. I mean, you check all with KRAS G12D mutation. Have you checked with other kind of mutation of KRAS? Because it's quite known that depends on the mutation, the answer of the therapy is different. I mean, do you expect that with different mutations, the same results?
Yes, we do. So what we know is that particular cell state is driven by overactivation of the MAP kinase pathway. So we have similar data with other G12D inhibitors. We have similar data with BRAF inhibitors in collaboration with Hector Palmer. So these BRAF tumors also are enriched in the oncofetal EMP1 state. And when we treat them with BRAF inhibitors, they switch into the LGR5 state. There are 3 other papers with similar data coming up. There is one paper in Nature that just came up by Owen Sansom, similar data. There's another paper by Fred Savage, I hope it gets published soon. Basically, it's a G12C inhibitor doing the same stuff. And so we think, yes, that essentially that is just about overactivating MAP kinase signaling upstream.
Okay. Well, thanks again. Really, really nice work.
Thank you.
There is, we have a coffee break. I think it's roughly 30 minutes. I will make some noise outside to try to bring you back in. What do I have to do? Coffee break. Okay. Next session, 11:45. Good. Very good. Okay.
Good morning, everyone. Thank you very much for having me today, and thank you also for giving us the opportunity to explain our work that we are doing here with the data that CNAG is generated, genomic data. So this data that we are working with CNAG is in the context of 2 core research areas in our laboratory of the Hereditary Cancer Genetics Group in the Vall d'Hebron Institute of Oncology here in Barcelona. These 2 areas is on doing research on genetic susceptibility to hereditary breast and ovarian cancer and genetic susceptibility to radiotherapy response. So I will give you a scope of the research that we are doing in these 2 areas. So in the genetic susceptibility to breast and ovarian cancer, before explaining the projects, I will give you a brief introduction. I'm sure that you are very aware about the genetic underlying susceptibility of the breast cancer risk of developing breast cancer over the time. Is consistent of mainly 3 group of genes in the very well-known that are BRC1 and BRC2. Pathogenic variants in these genes and together with others confers a high risk of developing breast and ovarian cancer over the time. This group of genes are named as high penetrant genes. So then the second group of genes that we have, that we know, they are conferring a moderate high risk, lower than those that are conferring by the high risk, and are consistent also of other DNA repair genes as ATM, CHEK2, RAD51, RAD51C, D. And then the third group that we know that are conferring the risk of developing breast cancer over the time are a group of hundreds of different common alleles that are across of the genome, and by itself alone, they are conferring very, very low risk, but the aggregation, doing a polygenic risk score that we call polygenic risk score, confer a lower risk, and that's why we call the lower predisposition to developing breast cancer. So in the clinic, now in the genetic testing labs, the genetic diagnosis of this disorder is done through the screening of a panel, a bunch of genes of penetrant, high penetrant, and moderate risk only. So the benefits of having a positive genetic diagnosis, meaning we identified a clear pathogenic variant in this bunch of genes, is because the affected individuals can be opt for more tailored treatment. For example, the PARP inhibitors that is specifically for BRC1 and BRC2 tumors, They can also, the affected individuals can also have a reduced future risk for a second breast cancer or for ovarian cancer. So for healthy carriers, they can opt for a more breast cancer screening for early diagnosis, and also they can opt for preventive surgery, mastectomy and oophorectomy. However, in the clinical testing, more than 50% of the patients that are routinely screened, they don't have a positive diagnosis, a genetic diagnosis, meaning that we don't detect the pathogenic variants. So in the lab, we think that this missing heritability is caused by three theories, hypotheses that we have. The first one is due to the genetic test limitations, that is the gold standard now in the clinics is doing this only analysis of this panel of genes. And in DNA, so maybe the genetic test limitations of this test we can miss pathogenic variants. The second one is maybe we don't know yet the complete genetic architecture underlying this disorder. So we know we need to discover new genes. And the third is the large list of variants of unknown significance that can be classified either as benign or pathogenic, but with only the DNA information, we are not able to classify in these two groups. So today, I only present the work that we are doing regarding the first hypothesis that we have, the limitations of the gold standard running genetic test that now is used in the hospitals. So for that, we addressed this limitation, evaluating the role of whole genome sequencing and germline diagnosis for hereditary breast and ovarian cancer. This project is under two granted projects, one from the Instituto de Salud Carlos III and the other from the Breast Cancer Research Foundation. So the investigators involved, some of BHIO, many investigators of our hereditary cancer genetics group, and from this house, the investigators involved are Raoul Tonda and Jean-Remy Paul Trotta. So the objectives that we have with this project is to compare the diagnostic yield between the whole genome sequencing and the hereditary breast and ovarian cancer panel that is run and routinely done, determine the added value of whole genome sequencing over single gene test for cascade analysis, assess the secondary findings different than the hereditary breast and ovarian cancer genes detected by whole genome sequencing, evaluate the clinical feasibility and genetic counseling, very important, for whole genome sequencing, validate the accuracy of whole genome sequencing for genotyping low-risk variants used in the polygenic risk scores estimations. And finally, with all this data that we want to gain at the end of the project, to do a cost-effectiveness analysis to evaluate the budget impact of having whole genome sequencing in the clinical health system. So for addressing that, we are included, and we want to include around 500 patients in two cohorts. One is the panel testing, patients that are referring to the clinical genetic lab to test this panel of genes, and the other cohort is the cascade testing. All this study is done prospectively and blinded, meaning that the results that we got from whole genome sequencing in the lab we are not aware of the results that are getting during the routine lab test. So the methodology is extract DNA from whole blood, Illumina short read, and 30 pair of coverage. So I will share with you some preliminary data, the first data that we have, because the project is not finished yet, regarding to the comparison between whole genome sequencing and the panel. We saw that we got a high concordance of the variants, regardless the effect they have. We wanted to answer the question is, if we want to replace the genetic testing in the lab we need to confirm and to demonstrate that the whole genome sequencing is detecting at least the same variants that are detecting in the genetic lab with the other techniques. So answering this question is that almost 90% of the variants are the same between the two techniques, and infrequently, around 2% of the variants detected in the whole genome sequencing is unique for this technique, and around 4% is unique for clinical panel testing. We have also identified for the first 100 samples two deletions, BRC1 in BRC1 and P10, and one duplication. We need, at the end of the study, we will classify these variants as benign or pathogenic and compare with the panel testing, if the panel testing is detecting the same alterations. So we wanted also to answer this question: is the whole genome sequencing accurately genotyping 40 SNPs and 300 SNPs that are needed to compute the polygenic risk score for ovarian cancer risk estimation? So for the first 135 samples, we saw that almost less than 1% of the SNPs fail at least one of the metrics, mainly the red depth, strand odds ratio, the depth breadth, together with genotypic quality. So it seems that we could, at the end of the study, that the metric that we want to establish is which is the thresholds to consider a true cold SNP, to diminish the possibility to have false positives, okay? So with this first data, we are providing metrics and data that the whole genome sequencing can replace with a high accuracy the panel testing routinely used currently in the labs. So in the same hypothesis that we are limitations of the DNA panel testing, we are missing the variants, we hypothesized that the blood RNA sequencing can detect these missing pathogenic variants in families in which, after a DNA panel testing, we don't get a pathogenic variant, was inconclusive. So this is in the context of this Instituto de Salud Carlos III project. The cohorts included were three 45 hereditary versus ovarian cancer cases from families, highly suggestive of having a hereditary cancer, but after a DNA panel testing, we didn't identify any variant. 2 positive controls, meaning that they have already known, characterized in the lab, RNA alterations, and 6 healthy samples. So this RNA sequencing based diagnosis is done in collaboration with Anne Esteve from this house, from the functional genomics team, and the methodologies, as you know, extraction of RNA is whole blood. RNA is bulk short read sequencing using Illumina, is with a coverage of 150 per. And the analysis, the subsequent bioinformatic analysis, is done by Anne Esteve and her team, and is based on this detection of RNA outliers pipeline, known with the abbreviation DROP. So the idea is to use this pipeline, this bioinformatic DROP pipeline, to interrogate the panel of genes that is analyzing DNA, but with the RNA sequencing data. Okay, so this is an example that how the DROP outcomes. Here we can see the outliers of low expressed genes that can be underlying the disease. Okay, that needs to be confirmed with orthogonal RNA tests. Another outliers that this DROP pipeline give is aberrant splicing, and one of the first results that we got is an exon skipping of the exon 9 in the ATM gene. Here in the the so-called sashimi plot, we can see that in the case of the hereditary breast and ovarian cancer, there are 73 reads indicating the skipping of this exon 9 compared to 12 reads in a healthy controls. So this is from RNA-seq analysis, and we, with a different technique, with orthogonal technique in the lab, we confirm that this is real, this is skipping of the exon 9, and it seems that is complete. So it could be of clinical significance for that family, but this needs to be, this completeness of the splicing needs to be confirmed. So this is projects that are ongoing. So we wanted to share, despite they are not finished, but to give you a scope, the things that we are doing in collaboration with CNAG. So now shifting to the second area of researching that we are doing in the lab, regarding to the genetic germline susceptibility to radiotherapy response. So a complete, not so completely different, but different to hereditary breast and ovarian cancer. So the radiotherapy is clear, the role that has in the treatment, in cancer treatment, increase the survival of the patients, and decrease the local recurrence of the tumor. So more than 50% of the patients are treated in one moment of their journey of the cancer treatment with radiotherapy. However, a subgroup of patients can have secondary effects due to their extreme sensitivity to irradiation. In breast cancer, the late effect, toxic effects, can have around the normal tissue that is irradiated together with the tumor, and the patients can suffer of fibrosis, telangiectasias, atrophias, and more than 10 years after radiotherapy, the patients can suffer for cardiac toxicity or secondary malignancies. So the ideal situation for the radiation oncologists in the clinic would be to know before radiotherapy, before giving the radiotherapy, to know what patients are at risk of developing these secondary effects, to adapt the treatments. So far, despite the work that we have done in the last 10 years, there is not an optimization of radiotherapy based on genetic markers, biological markers, due to a lack of clinical validation of these potential candidates to be a good biomarkers of radiosensitivity. To address this unmet need, we have participated in the last 10 years in different European projects. This first, with the acronym REQUITE, was aimed to do a collection of data, clinical data, patient data, before and after radiotherapy for around 4,700 cancer, breast, prostate, and lung cancer patients across, recruited across different European centers and US hospitals, and with a standardized collection of data and follow-up after radiotherapy. And also we collected DNA and RNA before radiotherapy. The second subsequent project, European project that we were involved is named RAD-Precise, and the goal was to work with the samples and the clinical data that we collected in REQUITE to build predictive models and to implement these predictive models in the radiotherapy treatment planning systems. In this project, in RAD-Precise, we led the work package 3 that was aimed to identify differential expressed microRNAs as novel biomarkers. So for doing that, we did the study in 2 stages, discovery and validation. In the discovery, either in the discovery or the validation, we match patients, breast cancer patients and prostate cancer patients with and without toxicity. So for the discovery, the RNA extract, the whole blood RNA is not plasma, is whole blood RNA. The RNA was isolated before radiotherapy, and in the discovery phase, we sequenced the small populations of RNAs using Illumina microRNA sequencing. And the top candidates differential expressed in the discovery were validated in a second stage in the validation using a different technique, nanostring. So we got only significant results for breast cancer patients, and in which in the discovery, we got a list of 90 microRNAs with a significant adjusted p-value that were differentially expressed. And with that, with the heat map, we could distinguish more or less two groups, cases and controls, toxicity versus no toxicity patients. And this is the volcano plot showing the list of the 90 microRNAs with a significant adjusted p-value. So then we wanted to know these 90 microRNAs were in which pathways they were involved, clustering the microRNAs using gene set regime analysis. And we saw that they were mainly grouped in transcription, DNA repair, cellular senescence. So pathways that make sense to that participate in the response of the cells after being irradiated. So in the validation, we selected 22 to be validated using NanoString, and from 4 out of these 22 were validated, and the process that these 4 microRNAs were involved were in fibrosis, inflammation, and apoptosis, reinforcing their utility as biomarkers of radiation toxicity. So with this validation in independent cohorts, we think that these 4 microRNA signature can have a potential patentability. So for that, we asked for a grant, and thanks to this project, we are now finishing the whole story, let's say, because we are now doing the predictive models. Because one thing was to demonstrate that we're different. These 4 microRNAs were validated in independent cohorts, but now we need to demonstrate that together with the dosimetric clinical information and patient tumor information can have, plus these 4 microRNAs, can have a good accuracy to classify the patients having or not toxicity. If it's true, we will go for the patent. So for closing the presentation, I like to close with this, that summarize our expertise in the lab is to translate germline genomics into precision cancer prevention and personalized radiotherapy. So it's the time of acknowledgement. This is my laboratory team in the hereditary cancer genetics group, and acknowledge that Setare and Laya are doing their PhDs, their doctoral thesis in the whole genome sequencing analysis of hereditary breast and ovarian cancer genes, and the former predoctoral investigator, already a PhD, Esther Aguado, who was the responsible of the work of the microRNA signature and radiotherapy response project. But we are part of the bigger group of the hereditary cancer genetics group, led by Dr. Judith Malmaña, who is here, and all members of the group. Without their implication and dedication, all these projects weren't possible. And also to acknowledge the radiotherapy project collaborators, especially the radiation oncology department in Val-d'Hebron, the collaborators in the European project, REQUITE and RadPrecise, and also the work done here in CNAG. And finally, a big thanks to patients because they generously shared their data and samples. And thanks for your attention. Thank you.
Thank you very much, Sarah. Questions? It's a delight to see that students who went through the lab end up in nice places to work. Marianka.
Thank you, Sarah. That was super interesting. If there's time, I have two unrelated questions. Let me start with your last part of your presentation. So you only found your microRNAs in breast cancer. So is that a power issue, or do you have other evidence that they're only relevant in breast cancer?
Yeah, we have several hypotheses about that because with prostate cancer, we didn't have this signature, these differential expression genes. There are several possible reasons. We haven't done any experiment to confirm that, but first is that the toxicity events in prostate are varied. So maybe there is a limitation in the size for the different side effects. And in breast compared to breast cancer, that there were only clustered by two very clear toxicity, that is fibrosis and atrophia. That is one. And the other thing also is that maybe the T lymphocytes that react to inflammation to the skin have a very specific role compared to the gastrointestinal or urinary toxicity. But this is our hypothesis, but we don't know yet.
May I ask this? Sure. Yeah. Okay. So I was also interested, you're going to do this project for ovarian cancer, where you're going to look whether whole genome sequence data is suitable to derive polygenic risk scores, right?
Yes.
So I was wondering, because one of the things we're running into trying to bring this into the clinic is that it's often difficult to genotype every single variant. So I was wondering what your view on that is, and also the use of imputation to a reference genome.
Yeah, so with the whole genome sequencing, fortunately, we don't need to impute it because we are detecting all the SNPs. The thing here is to establish, it seems, at least for the first 135 samples, that the SNPs fail one of the metrics. We are analyzing 5 or 6 different metrics of the sequencing coverage, sequencing final effect, final call based on the sequencing. So it seems that we are going to cover and to genotype all the SNPs. So that is the utility to do it through whole genome sequencing. So we need to demonstrate, but we are in the lab not only for ovarian cancer risk, we are going to do the same analysis for breast, the 300 SNPs, yes, and prostate and maybe colon. That is the utility, in fact, of the whole genome sequencing, that you can address and expand the question that you have regarding to the polygenic risk score because you have the data. The only thing that we want to give is the metrics. How good is a genotype called using whole genome sequencing with 30 pair to say is a real genotype?
I would be quite confident that you can get all of those. It's just, I mean, it depends on where you set the threshold.
Exactly.
It's usually quite good. And I mean, these aren't difficult to genotype variants.
Yeah, so that is, yes, this is a good point, but there is not a formal analysis published so far. So that it could be, because if you need to demonstrate that you do with good accuracy and performance, I think it's better to say, yes, this is the values, this is the, yeah.
Okay, thanks ever so much.
Thank you very much.
I have a question.
Oh, another question. Wait.
Thanks for the presentation. I want to ask about the genomic integrity, about all the tumor samples that you work. You talk about SNVs and small genomic rearrangements, and it is known that the large genomic rearrangements as well could affect in the gene expression and all these things. So I want to know your point of view about what happens with the structural variants in all these contexts, because the missing heritability could be explained as well, these large genomic rearrangements.
Yeah, you're absolutely right, but only to clarify, we are working with DNA extracted for germinal blood. So the quality of DNA is quite good. Sometimes we have some surprises. So the DNA, we are not working with tumor DNA, so that problem we don't have. But it's true that for copy number variants, structural variants, the quality of DNA needs to be high. So the tumor DNA is a problem, especially extracted from paraffin samples. But we don't have this problem. And one of the potential value of the whole genome sequencing is just, as you said, addressing identifying these structural and copy number variants. Yes.
Another question. You're getting a lot of questions. That's good.
Yeah.
So thank you very much for your presentation. My question is regarding just the last one. So it's not so important, it's important the quality, but also the method that you are using for NGS, for reading your DNA. So using SOREAD, basically, you are basically losing a lot of structural variation. Are you planning to do some kind of analysis with long reads just to be sure that you are not losing most of the structural variation, considering that you have a very high quality DNA?
Yeah, in the RNA-seq project for the negative families, that is not a very nice name, but we usually said the inconclusive DNA testing, families that are highly suspicious to be a hereditary cancer, we are doing this RNA blood analysis. For that bunch of families, we are going to do long read sequencing, trying to not to lose all the structural variants. Yes.
Great.
Okay.
Thanks so much.
Thank you very much.
Okay, so. So the next presentation is by Matteo Spagnoletti. So he's from Roche. He's going to show us a new sequencing machine. Just, I don't know, to the right. I'm not sure.
So hello, everyone. Thanks to CNAG for inviting me, and thanks for allowing Roche to be partner of this great event. So my name is Matteo Spagnoletti. I'm the area manager for the launch of a new sequencer for Spain and Italy. And today, I'm really excited to give you a very quick overview, just 15 minutes, about sequencing by expansion technology and what Roche is doing for the launch of the sequencer in development. Well, Roche is not new to the sequencing ecosystem. I think the company requires a lot of presentations with more than 100 years on the market, and we have already plenty of products in our sequencing portfolio for genomics, including automation, library prep, and data analysis solutions. However, what is notably lacking into this space to complete an end-to-end solution is a sequencing instrument, is a machine. So Roche is planning to fill this gap next year with the launch of the Axelios One platform in 2026. We unveiled sequencing by expansion earlier this year, but actually has been many years in the making. So this is coming actually from bringing together two powerful technologies. So one is the chemistry itself, is sequencing by expansion chemistry acquired from Stratos Genomics in 2020. And the second piece is a high throughput sensor array coming from Genia Technologies that was acquired earlier in 2014. Bringing together these two technologies really allows Roche to move into the future with a differentiated platform that can serve the needs of the growing NGS market. But let me say how the workflow works, how sequencing with the SBX works. So there are three main steps. One is library preparation, very similar to other sequencing technologies. But then there are two steps that are separated for flexibility. So one is the synthesis of expandomer molecules, and I will tell you a bit more what they are. And this is happening on a synthesis instrument. And then you have single molecule sequencing on a CMOS-based sensor molecule that is happening on the sequencing instrument, separated from the synthesis. So you have this flexibility, and then also, of course, you include data analysis. Let me tell you a bit more about what SBX is for real. So the process works by converting native DNA into a much larger and bulkier synthetic polymer. So when you take your library pool, every molecule in your library get converted into these molecules that we call expandomers that are 50x larger than native DNA, but conserving the same sequencing information. So every molecule, every read in your library essentially is done by expandable nucleotide triphosphates, or XNTPs. These are really the elegant building blocks of XBX. And if you look at this image from the bottom of the image going up, so every XNTP is built of different components biochemically. So you have a region that allows the expansion. So you have a cleavable bond that allows the molecule to extend. You have an enhancer region that allows the polymerase to incorporate these very special molecules. You have a reporter code that is very important because it's actually the area that encodes the base information. So for the base call and recognize the 4 different bases. And also on the top, this is a very clever engineering point where we have included a translocation control element. That's the name of this part of the molecule that essentially blocks the XNTPs into the pore when the translocation is happening, allowing stochastic translocation not to happen, but being deterministic. It means that you don't have multiple nucleotides passing through the pore, but you can really control the pace of the sequencing base by base. The expandometer synthesis is actually happening on the synthesis instrument. So you need sequencing chips and sequencing regions, and you can do up to 4 library pools in one run of 4 hours. Essentially what is happening chemically is on the flow cell, there is this extension oligos on the synthesis chip. The library flows through the chip. It hybridizes to the oligos. Then you have the polymerase coming in. It's a very special polymerase that was engineered with more than 40 mutations to extend and create these expandometer molecules. And when the read is complete, then it gets extended and photocleaved and then prepared for the speedy synthesis to the sequencing step. So the sequencing instrument is really where the CMOS sensor module is holding. So this has a dense array of 8 million microwells that can really lead to unparalleled throughput and pore-based sequencing. So here you see an image of each of these 8 million pores. And it can sequence as much as you like from 10 minutes up to 4 hours. So you can really decide how much data you want to get out of the chip. And it's really, really fast with this amazing throughput of 500 million bases per second. So really incredible what we have been doing. But another characteristic of the chip is that the chip is reusable. So for every run, you can clean up the chip, like remove the bilayer and rebuild it to allow a very cost-efficient and flexible process for sequencing again. So again, reusability is one strong point. The other one is also the controlled translocation. I want to stress you don't have stochastic passage through the pore, but you are really controlling base by base the bases that pass through, and these are controlled by electrical pulse to register the correct base calling. On top of that, we also worked on flexibility on the library side. So SBX allows 2 library structures and workflows, 2 main workflows. One is called SBX duplex, and the other one is called SBX simplex. So the duplex workflow is built to enable high accuracy. So really look at future applications like whole genome sequencing, exome, somatic oncology, MRD, everything where you really need the highest quality of your data. And this is achieved by hairpinning the 2 parental strands of your library when you come to the library prep. And this allows to pass the same molecule twice through the pore, so you can do intramolecular consensus and boost base accuracy. On the other hand, the simplex workflow is trading the highest quality of the data for either a ultra high throughput or read length. So here is really, if you want to use short read libraries, you can really generate an humongous amount of data for proteomics, fragmentomics, spatial, everything you need, like a high number of reads, or you can sequence longer reads. So the workflow is already able to give you more than 1,500 base pairs at launch, but we think there is headroom for making even better than that. So applications where you want to look at structural variants, phasing, RNA isoforms can get really a benefit of having longer reads as well. So very flexible. So I don't have time to go a lot into the details of the data quality, but I want to stress maybe one aspect that was less visible in the initial presentations versus more classic like quality metrics like PRED scores, is the SBX level of accuracy in variance with length. So this technology really allows you to have quality that is exactly the same across the whole read. So the technology is very clean, the signal is very clean across the whole read length, and this is notably valid also for the simplex workflow. So you have reads that are 1,500 bases that are having all the same quality across the read. Potential use of SBX are many. So Roche has a clear direction where it wants to go, but it's launching this platform as an RUO, research use only open platform, and we think that there is space for so many different applications. So just you can think of what you can do with these instruments, really. Well, I want to show you just a couple of data sets from our early access collaborators into the oncology space. We have been collaborating with the Hartwig Medical Foundation. They do routine tumor normal whole genome sequencing for routine cancer and diagnostics and research. So Edwin Kuppen team has been taking one of the prototypes to do early access, and they generated a data set of 30 tumor normal genomes with SPX, comparing to the routine pipeline. So here you can see the two data sets, one in front of the other. And what we can say, they presented, Edwin presented more in detail at AGBT, if you want to know more about what they have been doing with the SPX prototype. But what you can see is really an incredible level of consistency with the routine pipeline, where most, if not all, differences were explained by stochastic effects involving real low variant allele frequency of subclonal variants, so the data are almost identical for SNVs. And the empirical platform single nucleotide variation error rate for SBX was even like lower, like 1.7 lower than the routine pipeline. So very strong data set, high quality of data available for research. And SBX also show very strong homopolymer performance and very strong performance on FFPE samples as well. We are also developing internally minimal residual disease whole genome sequencing workflow. Of course, you can input your data. Here is just what Marco Corris' group is doing. It's still in development. But you can see how the pipeline works. It takes essentially the SBX-D reads in with already very high accuracy on the data, and it boosts the accuracy up by doing different steps in the pipeline, including aligning to the reference genome, then doing clustering and positional collapsing with intermolecular consensus, boosting accuracy even up, and a final step of read filtering that brings the SNP accuracy above Q50. So here is where you can really go then into the tumor fraction estimation. And the group have done an early experiment with 15 cell-free DNA and 15 healthy samples. There were actually very challenging samples in this cohort, like early stages tumor, different cancer types, but sequencing 100x with SBS allowed to determine minimal residual disease even on the most challenging sample, where the tumor fraction was as low as 1 × 10⁻⁶. So strong data set with SBX, just to show the quality of the data that you can input, for example, into an MRD pipeline here. And moving out of the oncology space, I want just to spend a couple of words about what the machine can do on the RNA application. We have partnered with Aziz's group, Methods Lab at Broad Institute, to do some early testing with this technology. Here you see two scenarios, both with 10x libraries. The first one is a 1 million cell perturb-seq experiment where they have been using more than 400 cell lines. So we are very excited to actually see the data out of this and results that they are getting. And for this run, they did a 4-hour run with SBX where they generated more than 30 billion reads of cell ranger barcodes, so very short library reads that were mapping consistently to the reference probes. So really an incredible amount of data. And conversely, doing 5' gene expression, still with 10x, they have been instead using the power of the longer reads to do isoform detection, where they have been able to generate in 1 hour 3.9 billion reads, where 200 million reads were above 1,000 base pairs. So again, a lot of flexibility here on what you can do with the machine. To conclude, just want to say that SBX technology is really designed for flexibility and performance and really looking at how you can scale into the future with your experiment. You have flexible operation, sequencing across the whole spectrum. You have accuracy of the data with the SBXD workflow. You have this amazing throughput. It can do essentially whole genome sequencing of 7 samples. They did it with Genome in a bottle in 1 hour, 30x. And then you have the read length flexibility. So really duplex with short reads or simplex with even longer reads. But one aspect that I did not have the time to cover is time to result. Like there is a version of the SBXD workflow that is modified to accelerate the library prep step to the point where you can do sample to VCF in less than 4 hours. Actually, we even advertise this as the Guinness World Record online. But I think aside from the marketing aspects of that, I think it's important to reiterate how this is important for specific applications like NICU or difficult samples where you need the results immediately. Last but not least, cost efficiency. I think it's important to say the system is built to reduce costs on sequencing and really allow you to work well on your research. And yeah, that's all from me today, and I hope you enjoyed, and let me know if you have questions. Thank you.
Great. Thanks so much. Time for one question. There, look, you have one. Stay close to the microphone.
Nice presentation, thank you. I have a question. Maybe you say in the slides, but I would like to know about which is the resolution that you can get with this technology. I mean that the variant allele frequency is below 0.001, for example. You can detect these small subpopulations, for example, in cancer. I say that because there is a tool called Dragon that uses an alignment, quantic alignment mapping strategy that's able to decrease the detection of really, really low frequency variants in the samples. I just like to know if you benchmark something in this context.
Well, thank you for the question. I think I'm somehow not allowed to deep dive into, let's say, technical questions on the data, but I'm more than happy to bring you people that can do that for me. I think that what you said is right. Like, it depends on the specific algorithms that allow the technology to boost the quality. The SBX technology can really give you the highest quality of data that are now on the market. So I think if you can do it with other technology, you can definitely do it with SBX. But let me take it offline and help you to understand a bit more about the details you want to cover, okay?
Okay, thank you.
Okay, thanks ever so much, Matteo. Okay. So it's my pleasure to introduce Nadia Sadliarova from Illumina. So please.
Thank you very much. I think I can just, okay, I'm not touching anything. Thanks a lot for inviting us to be the part of this meeting. It's really a big pleasure. And I know that Illumina has been supporting this meeting for quite some years now, but it's my very first time, so I'm super excited to reconnect with the community. I'm Nadia from Illumina, and I know the pressure is high. I'm the last talk before the lunch break, so I have to keep it more concise. I was told, with no further ado, let's hit the road. And today I'm going to speak with you about the developments from Illumina's side that we are making in the space of the multiomics. And this meeting has been a wonderful example of how such complex diseases like cancer requires very complex approach and the ability to look at the different layers. And therefore, our commitment as a company for multiomics is really the area where we see that the more effort we put in the field will allow us to unlock unprecedented depths of biological insight by enabling the community to combine the information from very different layers, all the way from the genome, for example, with the description of the prognostics groups, all the way to the phenotype with the outread and the, for example, proteomics level. And a bit of the personal note, like for me as a person, before joining Illumina, I've spent many years in the field of the academic research on the side of the transcriptomics, doing my PhD and postdoc. And I was focusing on the side of the RNA. But today it's super exciting to see how NGS has become really the readout for many different omics and also allowing us to look across the modalities. So, and this is like, currently the big focus for Illumina, and what we are building from our side. So historically, the researchers have been limited in a way of the need to choose like, between the, really the type of the information that they are going to, pursue. The material was limited. The workflows were very complex. The overall price, to do the discovery was also the limiting factor. In a way, resulting to the need to really, limit yourself to be looking at one omics layer, like, for example, in bulk, and sometimes really compromising between the level of insights and the scalability. So this is what we are trying to actively overcome from Illumina side, so redefining the limits of what's possible, enabling the community to discover more with the whole breadth of multiomics and multimodal insight, providing the opportunity, of the end-to-end, but also like really flexible solution, maximizing their discovery power. And at the next slide, I will show you some of the most exciting, I think, the roadmap highlights. So in 2024, just a year ago, a little bit more, right now, Illumina has made several important announcements for the field of the proteomics, for the field of the single cell, as well as announcing the new ways of using the short-read sequencing information and bringing the constellation technology to light. And now we are at the end of the 2025, and I'm genuinely excited to see that, we, as a company, we made the announcement and we're delivering on the roadmap. So you see that some of the solutions have been launched already, like, the single cell, the way of advancing the proteomics, Illumina protein prep, as well as the Illumina 5-based solution. But then looking into the 2026, there are still quite some exciting essays. And in fact, today it was a bit of a challenge to say, okay, 10 minutes, what should I focus on? So I'm going to tell you, to give you a bit more of a glimpse in the two technologies, one that has been launched just a month ago, 1.5 months ago, Illumina 5-based solution, and the other one that is going to be launched next year, but this is the one that does not live on the slide, but is being co-developed with our beta testers, with the early access customers, and this is going towards the Illumina Special. So Illumina 5-based solution now available. Where does it coming to, and how does it prove the strategy that we chose as a company? So thinking about the DNA naturally, this is the multiomics molecule per se, because it brings the two level of the information on the side of the genetic variants that it might have, but also carrying those important epigenetic marks, like the methylation marks that define the property. And the assay, that we think will make a big difference, named Illumina 5-based solution, is the type of the assay that allow you with one shot get the both insights on the side of the genetic variants and the methylation component. And the way it has been enabled is based on the the efforts of the Illumina R&D team. So that's what we've been working on for several years by enabling the new way of the conversion chemistry. So the traditional NGS-based approach that you would use to detect the methylation are based those indirect conversion, where you would convert on the non-methylated Cs into Ts, resulting in the efficient way to approach the, the methylation. However, this treatment is known to be quite harsh to the molecule. It eventually very much skews the representation of the nucleotides, resulting in limited mapping efficiency, and therefore making it hard to simultaneously get to the point of the very accurate genomic variant detection. So what makes it different in case of Illumina approach now is that the key component and the key innovation of this solution is the direct one-step conversion. So essentially, the enzyme is doing the trick of converting the methylated Cs into Ts, and that brings in the result several important benefits that go towards the well-represented balanced library that doesn't have much problems on the side of the mapping efficiency, and allows you to get back to the high accuracy genomic variant detection while still being efficient on the side of the discriminating between the methylated nucleotides. And as a result, we're getting to this Illumina 5-based solution. So the assay, as I said, one assay that brings the both insight from the both omics layers at once. This is a very high-level slide, essentially just showing that we offer the whole workflow here, from the library preparation that is finished in less than 1 day, to the sequencing part, and of course, importantly, to the analysis with the 2 layers, the secondary analysis being enabled by Dragon, but also very important, the ability to combine the omics information with our newer released Illumina Connected Multi-Omics Tetri analysis software that allow you to combine very different omics and very different modalities. And of course, this is marketing slide. No one wants to look at it. Everyone wants to see the data. And here are some data snapshots. Again, in the interest of time, we won't have that much time to go through the details. But essentially, I want to make a couple of highlights and call-outs here, that the solution that we have extensively tested prior to the launch has been shown to deliver on par methylation detection with the gold standards, such as basulfide sequencing or EM-seq approach. At the same time, it provides superb genomic variant detection, so you can see the precision really hitting it compared to the other solution. And this is how the outlook of the output of the assay would look like, with both omics, both layers of the information being available for the view. And one more thing to add here that is, I think, very important is also, I started with spending a bit more time on the side of the enzyme and the what made the assay so special. And this mild conversion, the single-step enzymatic conversion, is in fact gentle enough to retain high complexity library for low input samples. And so here you see several data call out. So on the left top, we are showing the deduplicated coverage obtained from 600 million sequencing reads for different cfDNA inputs. And essentially, you can have already good feeling that it is compatible with very, very low input, starting from 1 nanogram of cfDNA, and expanding the ability to apply this approach into the applications of liquid biopsy, but also, for example, going towards the field of fragmentomics. We have several customers already kind of trying it out because the assay is now being officially launched. So there is quite some field experience from the peers, and I will share the QR code also for some of the recent presentation about customers having their hands on with the 5-base. But I know that I have like about maybe 3 minutes left to tell you about the second assay I called out. And this going now, changing the gears from the multi-omics approach with the 5-base that can be done on the whole genome, on the targeted area, or on the cfDNA, now going more towards the modalities. And again, this importance of and the value of spatial application is well recognized in this room, in this audience. Illumina has been a proud supporter of the spatial community efforts for many years together with our ecosystem partners, enabling the sequencing part. But one of the ideas of bringing the own kind of spatial solution was, again, coming back to the initial strategy to maximize the insight without compromising the scalability. And that's why we are very excited that in less than 6 months, we're expecting Illumina spatial solution to be fully launched. And in a nutshell, it allows you for unbiased discovery of the transcriptome, so the unbiased profiling of the transcriptome. It is a poly-A-based solution, so applicable for any eukaryotic types. It brings 4 times higher resolution compared to the on-market leading products. And poly-A capture enables detection of up to 2 times more gene than compared to the prop-based methods. There have been several important advancements and improvements done over the course of development of Illumina spatial technology. One of them goes towards the resolution. So we are speaking about the binning, in this case, within the 1 micron area. And of course, in order to go to the really subcellular picture, we have also to work also on the different parts of the solution, including the part of the subcellular fragmentation. So there are several iterations of the AA power cell segmentation improvements being still kind of, undergoing. This is one kind of aspect, very high resolution. But the other one that is pretty exciting and hopefully will also inspire many of you to think bigger in all the senses is that with this solution we are bringing the biggest tissue capture area. So what you see here on the slide is the actual capture area for the spatial slide that you would use when going with Illumina spatial approach. And its current dimensions is 1.5 to 5 centimeters, making this big, big area where you can simultaneously profile several tissue sections, which is quite exciting in order to imagine what, about the scale and how much you can do with the approach. And on the side of the workflow, we will not go into that many details, but I just wanted to make a call out that for this Illumina spatial technology, the idea is that we bring it without the requirement of an additional instrument, without the requirement of a special instrumentation. Of course, you would need some capacity of the histopathology lab by the ability of the section, the sample, to fix it, to stain, to image, but then the part after it, going through the gasket device that is very compact, and actually have it around here, so come by, I will kind of show you how it looks like. And then it goes through the permeabilization of the tissue, followed by the cDNA synthesis and standard library preparation. All the way to the Tetri analysis with the Illumina connected multiomics software piece that we are also making available to the community for the really user-friendly interface to visualize your finding. And the roadmap for the spatial technologies is quite rich. Of course, this is speaking about future is always exciting, and I'm going to be super excited in a year from now to see how far we're advancing it. So the current solution that is going to be launched is going to be for the fresh frozen tissues, but there is, on the roadmap, the compatibility with FFP, bringing in the enrichment panels, but also importantly, combining the spatial transcriptomics insight with the protein layers. And with this, I'm at the end of my teaser talk, so I hope that I could inspire you to think about some of the future and current application that might find a place in your research. At the latest American Society of Human Genomic meetings, our multiomics team has announced, has brought up this slide with those dots, the dark ones, being filled in this year from the side of the omics and the modalities layer, but you can see that those that are white, those are on the roadmap, and we hope to hear more about it soon. And with the last slide, I would like to thank everyone for your attention, and also, as I was calling out, there are quite some exciting resources, and most importantly, the presentation from the peers who have tried many of those technologies. So with this QR code, you get the access to the recordings and to the slide to the web pages with some of the files. And of course, we are around here, so I'm happy to take the conversations further to address your questions. And now I think that everyone will be excited about lunch going on. So thank you very much for your attention.
Thank you very much, Nadia. Does anyone have questions? Yes, no, maybe? No, I think everyone is sort of ready for lunch, right? Okay, good. I will make thanks ever so much again. I will make noise afterwards to try to get you back in, and then we continue after lunch. Bon appetit. Bon approvecho. Welcome back after lunch. I hope you enjoyed the food and everything. I hope you're all full. Not too sleepy after that, but we will try to keep you entertained. We'll try very hard. The first speaker after lunch is my colleague Sergi Beltran, who's been with me for how many years? 12 years. 12 years. So, yeah, he's going to show us new systems that you probably have not seen yet. So it's going to be interesting. Good.
Not that much. Okay, so good afternoon, and hope you had some good lunch today. And I was happy to see that there was still even some leftover food, so we can take over for the weekend. My only issue is that I had this food already Friday, and yesterday for the SMB, and today again. So others can have it today. Good. So I'm going to be giving an overview of some of the stuff that we've been working on or doing in the past few years. And the first thing I want to mention is that the bioinformatics unit comprises 4 main teams. The production bioinformatics team, led by Matt Ingham, which is the really working head-to-head next to I'm going to put my watch here so I see how long I take, next to the laboratory. And they take care of data management and quality control and transferring the data to the collaborators that we work with. Many of them are here, but also to internal teams that then do other types of analysis with the data. And they also developed the LIMS system, our laboratory information management system, which we use to track everything from start to end, and many of you interact when you have to send samples to us to enter the metadata about these samples. Then we have the variant calling and analysis team. This is led by Raul Tonda, and they mainly focus on developing tools or pipelines for analyzing data for variant calling, annotation. They do lots of marking, and they participate in many projects. You've seen a couple of them today, and you'll see another one afterwards. Then we have the data platforms and tools development. This is more an engineering team where, based on the requirements that we give them from the other teams, they help us develop very specific tools from a high-level engineering point of view. So it's more engineering than bioinformatics, actually, and this is led by David Episia. And then finally, the team I lead directly, which is the human genetics team, which is more focused on analysis and participating in research projects. So I did a bit of archaeology last night, and I digged up some slides from BCAST. and now Marianka is going like that. And then I had to keep going a bit years back into my presentations, and I found, I knew I had this slide somewhere. So this is when we started thinking how we would be organizing the different samples from the BICAS project, because she didn't mention, but they were coming from a couple of centers. They had to be QC'd, the slides, but also the extraction of the data. And early on, when they came to me with the press, like, no, we cannot do this with an Excel. It's 10,000 samples, normal per tumor. Many are going to be failing, so we need to have a system to track that. So very quickly, we set up a system, and it was done by Matt and Eloy in the production bioinformatics to take into account all the different characteristics from these samples and then pair it with a BCAC database and with our LIMS database. So we have some sort of integrated system for managing the data. So, just wanted to let you know that even though the science is exciting, sometimes also we have to, do things which are maybe not as fun as exciting, but they need to be done so that we can do the projects properly and we can track everything and don't lose our mind in the way. Also in BICAS, besides the development of the database, Raul's team and Mark Dabar also work or helped on designing the panel. They got into the Roche website and selected the probes for the panel. They developed the pipeline that then we used to be able to quickly analyze all this data, and they did the variant calling annotation that then was used for the analysis and results that Marianka showed earlier today. And because BICAS was such a large project with 10,000 tumor normal pairs for 20,000 samples, it didn't come to me as a surprise when I asked Raul, why don't you give me the numbers of the types of cancer that we've been working on in the last few years? And breast came up as a big letter when I put this into the workload. So I really had to downweight several times breast so that you would see the other ones. Okay, I had to downweight breast, I had to downweight colon, and also hereditary cancer. So we've heard quite a bit about breast today from Marianka and Sarah, and also afterwards I think from Conchi we'll be hearing from hereditary cancer and cancer of unknown primary origin from BioScan and Impact Genomic. And these are a few projects that we've been working on in the past years. And in Raul's team, as I mentioned, a lot of what they do is implement pipelines, but to do that, we do a lot of benchmarking. And this is just an extract of one of the tools that Jordi Morata has been recently testing to evaluate how it performs when we are comparing tumors and normals at different coverage. So you need to have tumor and normal at a certain coverage to be able to compare those and have good precision, good recall, and a good F score. One thing that is a bit strange here, it sounds look fun, is that even when we increase the coverage, we reduce a bit the precision at some points if we don't have enough coverage at some level. Okay, so that's an interesting observation, and they need to finish that work, and they've been doing that in many different tools, and hopefully a paper will come out of that with these comparisons. Then, once they have assessed tools, they put them into pipelines. We try to maximize as much as possible what is out there. This is a slide where you can see, and of course, Sarek here in the center of the slide, but also in red dots or red asterisks, you see models that we develop ourselves or that we add ourselves to these pipelines to add a bit more value into the data. For example, to compute some of the metrics that Marianka was showing before, and some of them we try to make them available also for broader use by the community. Okay, with that, I'm going to move more to the work that we've been doing in the human genetics team, with the collaboration, of course, from Raul's team and David's team also. And this is a project called SOVARDI, which is mostly on rare diseases, and some of you have heard partly this talk on rare diseases, mostly on neuromuscular disorders or intellectual disabilities, but I'm going to focus today on inherited cancer. So this is a project that we started, I think, 4 or 5 years ago, and it's a project that has a main aim to solve rare diseases. And we had 2 main approaches for that. One of them was to reanalyze thousands of data sets from patients that already had an exome or a genome sequence, but they didn't have yet a diagnosis out of that. And this was driven mainly by 4 ERNs: Ithaca, intellectual disabilities, NND, neuromuscular disorders, Genturis, inherited diseases, and RND, neurological disorders. Okay? So that was one of the approaches, reanalysis. And the other one was providing new analysis technologies for some of the data. Unfortunately for Genturis, which is the inherited cancers, we did some deep exome sequencing, which was useful, but also we tried to do RNA-seq, and at that time, or for some reasons, the samples that we got were quite degraded, and that data was not very useful. But I think Conchi afterwards maybe might show us some data that it's actually more useful from other projects. So in that project, we work with a distributed infrastructure. We put together, we encompassed all the data from these thousands of data sets through a system that we have developed in-house, which is called the RD Connect Genome Phenomics platform, and it's a PhenoStore module to capture all the sample and individual clinical and phenotypic information. Then we did some analysis already in-house, and we put all the data also to the EGA, the European Genome Phenome Archive, where the rest of the partners from the consortium were able to suck this data and put it either into a shared cloud space or into their own authorized local cluster to do other analysis. And therefore, we try to give a very comprehensive look at all of this data that had not been yet proven to be useful for diagnosis. We worked in a two-level expert approach. On one hand, we had a data analysis task force that brought in the bioinformatics experts, and this is mostly where my team and I was involved with, and we were leading a few of these teams and the task force itself. And then we also had data interpretation task forces. We had a task force for each of the different ERNs participating in the working group, and then together we worked on different working groups and projects to try to crack some of these cases. This is the list of the working groups that we established. And I'm going to give examples of the first two ones, the SMB and Indel, which was led by Leslie Matalonga from my team, and the second one on CMBs, which was led by Stephen Laurie from my team and German Debita from Tübingen. And in the first thing I'm going to show you is how we were very quickly able to diagnose a few of these cases. I mentioned before, we put all the data into a system, we had it organized in a database, and we could very quickly query that data. So with the help of David's team, we set up a few scripts that were querying the APIs, and then we were gearing towards identifying very, verging towards specificity. So we were trying to identify very likely causative variants for the diseases. Okay, so we were not aiming for sensitivity, but rather specificity to try to have only a few cases that the data submitters had to review from these thousands of cases, and hopefully these were already good diagnoses, and we could already clean up a bit the amount of data that we had to analyze. These were the results from the first analysis. You don't have to look at the specific numbers. You have the numbers here from the 4 ERNs, but I think the important thing is the summary at the bottom. At that time, we looked at 275 probants. Most of them were singletons, okay? So there are some challenges with analyzing only singletons. And we focused on a list of 229 genes that the ERN Genturis gave to us for the analysis. With that very quick analysis, we basically went down to 30 variants only that had to be looked up by the interpreters on 30 probants, okay? On average, 1 per probant, but for many of them, they didn't have to look at anything because there was nothing that was highlighted. And from this, we were able to solve 3 cases, so only 1% of solve ratio, okay? So it's low, but was a quick step that you can do reiteratively every time that new evidence comes in, or every year or every second year. An example of a finding was this case, this 25-year-old female that had diffuse gastric cancer and had already been tested for P53 expression, and this was wild type. It didn't have any coexisting disorders or abnormalities. The exome had tested negative, and unfortunately she received treatment but was deceased. And with the reanalysis of the data, we were able to identify a mosaic mutation that was present at less than 18% of the frequency was less than 20%, and that was useful in the limits of what people were looking at. So when you want to look at those cases, you want to look at many different options to go down and be able to find these disease-causing variants. Other examples come from the CMB analysis, and this was work published a bit later in last year, 2024. This group, as I mentioned, was led by Steve Laurie from the team and German Davidov. And they used 3 different tools to systematically analyze all these thousands of exomes, exome depth, conifer, clean CMB. But I think that the added value, what they brought into the table, was how they managed to integrate all this data together between the different tools with the already identified likely pathogenic or interesting variants from the SMB analysis, but also with the patterns of consanguinity we had identified in these individuals from the data itself, and also those for the gene list that we were provided. And that was also a way that took a bit more than the SMB to set up, because it took quite a bit of back and forth with the ITFs to set this up, but that was quite successful also in terms of reanalysis throughput. You can see that each of these tools gives a different pattern of length of deletions and duplications. Okay, so each of them seem to be doing well or being catered for different lengths. So that was important to be able to use these different tools. This is the summary of the 51 index cases that were solved from the CMB analysis. Only 4 were from the Genturis ERN, and you can see that there are many more deletions than duplications. Okay, that might be a thing that they might be easier to interpret and detect also, because duplications, we never know, how many more duplications are there. Also, you can see that there is a difference in length. The duplications, if you see the bars at the top, they tend to be overall much larger than the deletions, which we'll see quite a few which are on the lower end of the spectrum. Also, you can see differences on what the different tools might be able to detect, at least with the settings that we tested ourselves on these tools. In summary, from the tumor risk syndromes, we analyzed 300 provinces, so a few more than what we had done with the SMB analysis initially in 2021. 116 of those had at least one CMB, so we had to look at a few more cases than what we had to look for the SMBs, and from this we solved 4 provinces, so a bit more than 1%. An example of that is this familial adenomatous polyposis. This is a family that you can see that the disease is running in the family, and that was a challenging case on the APC because it was quite a small deletion of only 200 base pairs. So, and this was only detected by one of the tools. It just reflects back on the slide I showed you just before. Another example is on a larger variant, a large deletion. This is again a family with diffuse gastric cancer. It's a family that the first individual from the family was diagnosed in 1993. And then exomes of several members had been sequenced over the years, but until we analyzed that in SolveRD, they also returned negative results. And this was, at the end, what we identified was a partial deletion of CDHA1 and Tango6, that region. There's a few kilobases. And on all the individuals that were affected, we saw this deletion, and you can see that most of the 3 tools we tested here identified this deletion. But also we saw the deletion in one individual which doesn't have the disease yet, might be a healthy carrier, and apparently this has been, incomplete inheritance has already been seen before. The importance of that, obviously, is that we were able to finish the diagnostic odyssey of that family. As I mentioned before, the first case diagnosed in 1993, the first proband tested for SMBs in 2003, so 20 years ago. The first exomes analysis were done in 2015, so again, almost 10 years ago. So this is really an odyssey for a family until we managed to crack this one and find the deletion. So these were SMB, CMB specific results. Now I'm going to go and talk about the overall results. This is the paper that we published earlier this year in Nature Medicine, which brings in the SMBs, the CMB analysis, but also other types of analysis that we've done after that. And I think that the key message of that is that we not only used 2, 3, 4 tools for analyzing this data, but we went up to 14 tools for analyzing this data. So we really tried to go down and looked at many different types of variants. And we used many of the tools that you see here, which are publicly available, but also we used tools which were developed by members of the consortium, such as de novo CNN from Christian Gillison's team, Squirrels from Peter Robinson's team, or CleanCMB from Stefan Osowski in Tübingen. Overall, in the results, we were able to diagnose with a systematic analysis approach up to 8.5% of cases, and with an ad hoc expert review, meaning on parallel approaches, about 4% of cases. So the reanalysis throughput, including those that I showed you before on SNBs and CNBs, was about 12.5%. However, in the cancer syndromes was a bit lower. You can see that we are down here on the green bar at the bottom, and we have, although we have many candidate genes, the ones which were proven to be pathogenic are less than on the other ERNs. Could be because they were already better analyzed, or it could be because there are other underlying causes for this detection, maybe unknown genes. The gene list also that we used there was also very small, 230 genes, so maybe there are also genes that are unknown or that were not included in this analysis. You can see that most of the, on the right, you can see that most of the variants are SNB and indels coding, so in principle could have been detected with regular analysis, but many of them were either mosaics or they were in low frequency, in low coverage regions that people had discarded because of quality issues. So there were several issues why these were not identified, or also because they are in genes that before they were not known to cause that pathology. But also there was quite a bit of findings in the mitochondrial DNA analysis, not for cancer, but other types of disorders, and also the CMBs and other types of structural variants. Again, as a summary here for the tumor-rich syndromes, on the overall analysis, we went and checked everything back again. These are nearly 30, 60 probands, and we managed to solve 11 probands, 10 with a systematic analysis, 1 with these alternative approaches, ad hoc review. That's about 3% of diagnosis rate, lower than the other ones, as I mentioned before. 2 of them as SNBs, one small insertion, 3 small deletions, and 4 CMBs, and 1 that we don't know because it was has not been reported to us. That obviously has importance for the follow-up of the patients and their families. You can do cascade testing of those. In some of them, you can also guide the management of the patients. These are examples of some of the families diagnosed within the project that then receive some of these follow-up actions. I was having a conversation from someone before, they asking us if, what about FAIR principles? What do we do with open science? And this is an example where all the data we have collected in SolveRD, we put out at the EGA and in the RD Connect GPEP for access, through controlled access, so that others can go and do the research on this, I think, unique data set, which has more than 27,000 individuals from rare disease patients, including cancer risk syndromes. Okay, I'm gonna shift gears now. I think I only have like a couple of minutes left. I'm gonna mention that, and this is I think what Ivo was referring to. I talked a lot about inherited cancer, but we're trying to bring not only inherited cancer, but also somatic analysis into a useful and user-friendly tools. And we had a project, it's a very curious project. It's a pre-procurement call from Instant NGS that made a call to be able to produce such systems. And we have been developing tools for the data analysis lot of that call and the data interpretation lot of that tool. And basically, this is what the evolution of the GPEP looks like. So we have established now a module that allows us to put pipelines into a very systematic and standardized way to run the pipelines and track what has been done with each of these data sets. And this is tweakable, and you can follow up the process on how the data is going through the pipelines. And also we've established a model for reporting that instead of having you to go and look at many different combinations of filters, you can predefine which filters you want to define, for example, from the Catálogo Genético in Spain, and then already have some automatic prioritization. We are working also on a tool, I'm not going to be presenting it today, to classify oncogenicity of variants. This work done by Claire Morgan in the team. And also we are working on fine-tuning how we report all these variants for a clinical setting. Then last but not least, one of the things that I started with management of data. I'm going to end up with management of data, but we need to evolve from systems that are very static or very strict, that only take care of genomics data, to more flexible systems. And through the DataSCAD project, we had the opportunity to develop a new framework for managing data. It's called BioDN. This was work mainly done by Etienne Jodry, who already left earlier this year. And this is a system that allows, and we are now putting it into action in TruthCTR, maybe Ivo will mention something about that, but it's a system that allows us to then upload and manage data from different experiments, from very diverse experiments. The objective here is that if we are able to have a good data management of different multi-omics experiments, which we are doing more and more, that also will make it easier for us to track, to integrate this data. We are developing tools for integrating data from different multi-omics, and they'll also push this to the AI. But we need the data somehow organized and available, know that this experiment, this RNA-seq, this genome, this proteome, this epigenome belong to the same individual, and have all the metrics from this data together with the phenotypic information to be able to move forward with this kind of approaches. So I think with this, I will, of course, thank you for your attention, the team that is doing the work that I presented today, many of you as collaborators who send the samples to work with us in the analysis, and also the partners that have worked with us in SolverD and other projects I mentioned today. So thanks a lot.
Thanks so much, Serge. Do we have questions? Yes, please.
Thank you very much for your talk. I was wondering something that is you have not presented, but for some applications, for example, for estimating PRS, we need whole genome sequencing data from normal population. So it's quite difficult to find a Spanish healthy population to do that. So I don't know if you are aware how is the future solution, because I know there are local effort, so there are data there in the centers locally and also from patients. So we were talking yesterday in the dinner that there are a lot of data in Spain, but it's scattered and it's locally. I don't know if there are any effort in the future to...
May I can give you a couple of insights into this. One of them is retrospective data, the other one is prospective data. Prospective data, start with that. Ivo is leading the sequencing here in Spain of the Spanish cohort from IMPACT, and this has been done through two projects, the IMPACT, I think it's called Cohortos now, or GeoImpact, and the Genome of Europe itself, okay? And this is going to be providing sequencing of what, 12,000 individuals? 11,000 genomes in 2020. In principle, from, I'm not going to say healthy, but general population, okay, from Spain with good representation of the different Spanish communities. And then also backwards, if we look backwards, there's also quite a bit of data already done in projects such as IMPACT, which are from disease, people with a disease, or with relatives that might be healthy. And this data so far, it's quite siloed. It has been some technical challenges to put this out. My take is that this data is going to go to the local EGA in Spain, but in the meantime, we have it, in the different sequencing centers in Navarra, Galicia ourselves, and we are trying to federate this data in one way or another. We have frequencies of this data that might be useful, but maybe for PRS, I think it's a bit challenging because you need the individual data for each of those. But once you have the data in the EGA, then you can download the BCFs, the FASTQ files, and you're granted access, then you might be able to use that data. Even more here in Catalonia, we have the GCAT project, which has about 900,000 individuals, genomes, which we sequence here at the CNAG. That also might be another place where you can look at that. Navarra has other 1,000, 2,000 genomes, so there is that out there. But currently, you have to go and contact everyone independent to be able to access that data.
For the Genome of Europe data for Spain, that is going to be put in one place in Madrid. So that's going to go into one place, and then how it's going to be served back to the research community, that I don't know yet, but it's a good start when you have everything in one place.
Thanks for the presentation. I like it so much. I have a question about how GPAPI is able to solve the variant uncertain significance. If you do some test on that.
Yeah, so currently we are not applying any additional annotation to the abuses. Okay, so basically you might be able to see when there has been a change from BUS to likely pathogenic, for example. However, we are now developing a system, and Claire sitting next to you is actually working on that, and hopefully we can bring a bit of light in the next symposium onto that.
Okay, and I have one question more about the structural variant detection. I see that just in the pipeline use MANTA. I would like to know why, because there are many others that could complement this. And the second one is how you depurate the false positive calls using detecting structural variants by short reads.
Yeah, so the first one on the amount of tools, at the end we had to make some editorial some decisions on what we're going to use and what people were able to run into their own machine. So it had both a scientific reasoning behind running the tools and also pragmatic reasons. However, there were, and I can't remember now which ones they evaluated, but Delhi and other ones where I know they were tested. And then at the end we decided to go for MANTA for comparison that we had done before and also for pragmatic reasons. In terms of the FP, that is a very challenging question. And when you look at the SVs, by eye, at the IGB, you see that many of them are false positives. And this is a big challenge. And that happens also with the CMBs. So even though that some tools, they give you some sort of quality metrics, or, Bayesian score or whatever on the quality, when you look at it, if you decide to make a clear cut at a certain level, you will see that on the top ones, you still have maybe 40% of false positives. And below the cut, you still have maybe 30% of true positives, right? So it is very challenging to analyze this data still nowadays, I think. So one thing that we've been doing, obviously, is to look for variants which are low in the population, so they are infrequent in the population. That is a way that you can filter a lot of artifacts. And another one is to, instead of trying to identify clear cuts for these scores, to rather work on percentiles or deciles, right? So, okay, these are the 33 top percent scores, the middle ones and the low ones. And with this, we can give an estimation that if you look at, 10, likely 6 of them will be positive. In the middle one, it's going to be 50-50. In the lower one, it's going to be maybe 3 to 7. Okay, so that's the approach we're taking now, but it's not a perfect approach, but it's a bit challenging. Hopefully, bringing in also different approaches, like what we've done with CMB, also helps to do with that. But as you see there, actually, when we bring in data, some variants called by only one tool might be really relevant. So the union is not really that important then.
Thank you.
The intersection, sorry.
The sequencing method is also
No, and the sequencing method. It's, I didn't talk today about it because we didn't have any successes with cancer samples, but we also applied PacBio sequencing to some of these individuals, 1,000 more or less, and there was quite a bit of success with structural variants. We didn't apply it. It was sequenced in Nijmegen.
No, I just want to add, because I did the structural variant detection for GCAD samples. And we combined many variant colors to detect the structural variants, and then we apply machine learning models for each structural variant type, and the performance was differently across different structural variant types. So this is the reason why I ask about MANTA, just MANTA, because some tools are able to detect better for a specific lens, for specific structural variant types. So maybe combining with machine learning could improve the performance.
Yeah, something we can think for the next iteration. There are still thousands to be solved, so
thank you.
And we are collecting many more.
Okay, thanks so much, Serge. I should say I spend together with a bunch of people from CNAG every Friday morning discussing structural variants and how to call them and coming of which technology. It is brutal. It is brutal. It's not easy. Anyone who tells you that structural variant calling is no problem is lying or hasn't done it yet. That's sort of the thing. Good. It's my pleasure to invite Vincenzo Lupo from Agilent up here to explain technology stuff from their side.
Thank you, Ivo, for the invitation. So it's very nice to be here to represent Agilent and to give you an update on the target enrichment approaches for NGS. Pointer work? Okay, perfect. So just a brief introduction on the Agilent NGS portfolio. As you may know, so we are working for target enrichment approaches for around 15 years far. and with the Sure Select technology. And now we have also configured a new chemistry, which is the Sure Max, and the Avida technology, which I will focus more on this one. So Agilent technology kits can be automated in both liquid handling platform Bravo and the walkaway Magnus NGS prep system. And we can also offer automation of sample QC by using different equipment for capillary electrophoresis as the tape station Femto Plus, more suitable for long read approaches, and the fragment analyzer. So we don't have a sequencer, which somehow could be also a benefit, right? Because we can be compatible, we're also compatible with our catalog panels, custom panel, with a different sequencer for both short reads and long reads platforms. There are several applications we can work with from hereditary cancer, hematological, and solid tumors. We have discussed several of those. And the one that the two key benefits of Agilent is the customization. So we can work with human and other species, starting from the catalog panels. We can use the same catalog panels with the reduce or combine to add on a different content in our catalog panel. And most importantly, we can automate it in parallel, both DNA and RNA for the workflow of the library preparation. This is an example of our cancer CGP assay that's combined different biomarkers for SNV, CNV detection, and also for the homologous recombination deficiency detection, and the gen fusion by using in combination DNA RNA. This panel comprises more than 600 genes, and the interesting part of this is that we can actually reduce the number of genes by selecting specific cancer assay or increasing the number of genes by combining with new content. And this panel or custom panel of this can be indeed automated in the two platform. And of course, in other platforms that are liquid handling and are compatible with our kit. So we have other two main benefits that actually will be the main topic of this presentation, which is enabling target long-read applications that are also addressing cancer genomics, in particular hereditary cancer diseases, and also hematological diseases for the detection of, for instance, of the recombination for TCR and the IG recombinations. So, and the second part of the talk will more focus on the new technology of Avida, which enable the target enrichment for a multi-omic approach. So let's start with the first part of the presentation related to the target long-read applications. So the easy way of going on this is that we can, as I said, customize different panels, and for this reason we have decided to collaborate cooperatively with the two major vendors of long-read sequencing, which are Oxford Nanopore and PacBio, to address different applications and depending on the requirement of different laboratories. So the benefits of using target long-read target enrichment instead of using the native DNA or other DNA or RNA is that we can increase the coverage, which it's also very similarly important for detection of mosaicism of very low allele frequency variant, and in turn will also reduce the cost and that improves also the scalability of our samples in our laboratory. This is an example that we have recently published as a proof of concept. We have used our cancer CGP assay, around 3 megabases, for target enrichment approach. And we nicely could achieve N50, so an insert size around 5.4 kb, and with a nice also target. And in that case, 8 samples were sequenced in a promethion, achieving a coverage of around 500-600x. And this can be basically done by a modification of our Sure Select protocol, standard one with some modifications and then the steps. What we have to do is we have to share indeed the DNA Of course, we need the high molecular weight DNA to work with. And if we start from 8 to 10 kb, we can easily go from the pre-cap library to post-cap library to obtain this 5 to 5.5 kb that can really easily can be reproduced easily in the sequencing. As we are showing Oxford Nanopore, also we are also validated it in the PacBio system. Here I now show an example of a structural validation. I know that the structural validation is a hot topic here, but with the proof of concept, we can demonstrate that even for the very small fragments, but complex ones, because of their ATGC rich regions, long-read target approach can really improve the performance of the coverage of these regions. On the left side, we have an example of the exon 15 of the RB1, which is AT rich, and on the right side, we have an example of the hotspot promoter of TERT, which is usually one of the two mutations. It's an low coverage because of GC dropout for short reads, while when we sequence with that long read, we can really improve this coverage. Overall, the target-rich long read sequencing approach has several benefits. One of them is also to improve simple regions by just using the same panel that with the same probe boosting that we are using for short read panels. So let's move to the second part of the talk related to liquid biopsy solutions, related with the recently launched new target enrichment for Avida technologies. Here I would like to show as an introduction a couple of studies that demonstrated that the combining both genomics and epigenomics, in this case methylation analysis, is very important to advance for translational research, but can potentially be applied also for clinical applications because it's improved the sensitivity of the detection of ctDNA. On the left side is a work where they showed that they actually demonstrated that during the different time points of the follow-up of the cancer of CRC, the combination of both approaches really improve the ctDNA sensitivity. And on the right side, it showed that this is a case study, actually two case study, when the two similar follow-up the patients for the treatment, and one of the two patients presented with the BRCA1 mutations, germline mutations. However, they detected methylation's signature, hypermethylation on the BRCA1 promoter. Thus, confirming the importance of also including methylations in those cases also for hereditary cancer and not only the variant detection. But the real challenging of combining both approaches is the limited amount of samples. And basically, when you work with cfDNA, the limited amount of samples make it more complex, which in turn reduce the sensitivity. This is because of the conventional methods use a splitting of the samples, reducing the input, which in turn will reduce the sensitivity of the method. Avida technologies can address this challenge by being more sensitive. Here I would like to just show how Avida technology works. So this is different than the conventional method. This does not work with a direct hybridization of usually 120 base pairs biotinylated probes to the targets, but using two bridge probes that need to be specifically hybridized to the targets, otherwise there is no capture, where the anchor probes is then bind to these two anchor probes, improving the sensitivity and in particular the specificity of this hybridization. So we have three different workflows for the Avida approach. We can either use the Avida panels for the detection to increase the sensitivity of DNA variant by just using a DNA panel, or using a methyl panel. And the important things here is that we can use the dual workflow. So basically, we don't need to split the plasma samples, the cfDNA samples, and we can go through the protocol in 2 days by capturing from one side the DNA, the panel, that we can have commercial ones, or we can also have a custom one. And then with the uncaptured, we can still follow the protocol to capture the methylation markers. This method is based in the bisulfite soft method conversion, which is a bisulfite treatment that's happened after the enrichment, which is extremely important to do the conversion after the enrichment, and it's a bit conversion sulfite soft method, which is less damaging if we compare with the conventional bisulfite methods. Currently, we have three commercial panels, but we can also do custom panel. And then finally, we have this Avida methyl panel, which comprises 3400 DMR for the methylation signature. Agilent technology was selected in the OncoNGS European Consortium. So we are recently in the phase 3, so in the last phase of the contract, where with the other participant, we are now working in the validation study. So this is the last phase where the clinical validation is ongoing. And the prototype of this consortium was to address the best solution for precision cancer oncology by having an end-to-end solution. And for this reason, we have partnership with QCI from QIAGEN for the software analysis. And depending on the site, we are sequencing an Illumina platform in a AVT from Element Bioscience platform. For this project, depending on the laboratory needs, we have created 2 different panels. The OncoLB panel with 164 genes, and the OncoLB and the lymphoma panel with 86 genes, from which we're able to detect not only SNP, but also translocation and the copy number variations. And then we have a third panel, which is the combination of the 2 panel, and with the adding of not only the must-to-have genes, but also the nice-to-have genes. Here I would like to show the nice linear relationship between the cell-free DNA and the recovery of the molecules. So it's basically what we have seen, that there is a linearity between the input that you use and the recovery of the molecule that you get, meaning that we can give the dip that we need depending on the input, right? So in the end, for this case, we use the Avida DNA expanded cancer panel, and we can see that the correlation was very good for the both DNA panel and for the methyl panel. These data were generated using Seraker ctDNA, where we want to measure also the accuracy of the variant detection, and we can easily detect down to the 0.5%, which was the expected viral allele frequencies from the Seraker samples. But then we also spiked the ctDNA from Seraker sample to wild-type sample to get a bit lower allele frequency, and we were able to have a sensitivity of above 95% detection down to 0.201%. Although it's true that the allele frequency and the sensitivity also depend on the input we use. The more input we have, the most probability we have to get a low variant frequency variant. And this is an example of the Avida dual workflow. We are compare normal samples with the tumor samples. On the right side, you see the differentiated methylated targets as the score. So the methylated score that is compare normal versus tumor samples. And from the 6 to the 11, we have several tumor samples from lung cancers. And we were able to detect a 0.2, 0.3% mutation. However, for the number 10 and 11 samples, we didn't detect any variant. By contrast, we did detect a methylation signal, and which was significantly differences versus the normal control. So again, the importance of combining both genomics and epigenetics in the same methylation for the follow-up of the patients, for instance, also for MRD, and yeah, to also confirm the presence after treatment of the tumor. And this is, in particular, the result that we get from the AVITA method 3400 DMAR, where we can show that we can detect as low as 0.003%. We were able to differentiate from this to the 0, and with the still consistent of nice on-target and uniformity, and with a bit conversion, with a conversion rates of above 99%. And finally, so the DMAR targets of 3400 DMAR demonstrate to be differentially methylated. If we compare tumor tissues of the 16 tumor types compared to the normal tissue, and in the heat map, if you compare the purple one with the green one, so the tumor versus the normal, you can easily see how that it's easily to see the difference between the differential methylation signature. And this is my last slide. So to summarize, so AVITA Technologies, it offers first reduce turnaround time, so 2 days instead of other conventional methods, use more days. We can optimize the sequencing because of the target enrichment approach. We don't have to split the sample, so then we can detect both DNA and methylation, increasing the sensitivity. We can automate the system currently in the Bravo and soon also in the Magnis system, which is also it will reduce in turn also the turnaround time. And overall, we think that this is a very nice tool for the clinical research and potentially for the clinical applications. One last comment here is that I didn't mention that this is PCR-free, so we go directly from the input to the hybridization, which is also important for the recovery sites of the library preparation. So thanks the organizer for the invitation and you for your attention, and I will take any questions if there is any.
Thank you very much.
I was on time.
So if anyone has a question, we have it. Otherwise, we'll move straight on because we're quite tight. Thanks ever so much, Vincenzo. So we go, I can't see, I can see Conchi. Conchi Lazaro. You weren't in the right place, you were there before. Okay.
I move with this.
Okay.
My slides, I have to do something? Okay. First of all, thank you, Ivo, for the invitation. Congratulations to the whole CNAG family for your 15th anniversary. And it's a pleasure for me to explain here 2 national level projects dealing with addressing the cases unsolved of hereditary cancer. I have also to thank Sarah, Marianka, and Sergi, because the 3 here together give me a good introduction. I have a complementary introduction. I hope you are not get bored of hereditary cancer. But as most of you know, most of the cancers are sporadic due to the accumulation of several somatic mutations and the interaction of the several environmental factors. However, in some families, we do see clear clustering, and in a few proportions, we see like a Mendelian inheritance pattern, normally with incomplete penetrance. So this part of the pie that corresponds about to 5 to 10% of the cases, depending on the tumor, is what we call hereditary cancer. Hereditary cancer is the main object of study of hereditary cancer programs and genetic counseling units. Why? Because identifying patients that carry a predisposing mutation to cancer, cancer is not inherited, but the predisposition does, allow us to personalize risk, to offer different treatment, reproductive, and planning decision options to the carriers of these mutations. So in Catalonia, this has been ordered, although we were quite ordered before, but since 2021, the CatSalut launched what is called the Programa de Oncología de Precisió, and has different branches, and one of the branches was hereditary cancer. They create a group of experts that would work together with the whole community in Catalonia, deciding every year the indications for genetic testing and also the minimal number of genes that have to be studied. By this means, what the program wants, and I think it's doing it, is to provide equity in the whole territory of Catalonia. It happens the same in Spain, in different autonomous regions, and also now with the Cartera de Servicios of the government of Spain. The success in mutation rate depends on different parameters I will not enter, but the family history, if we study a patient with cancer or a relative with non-cancer, also the age of the cancer, and also the mutation rate depends on the technology, as you have seen previously. And always we have to deal with variants that we identify that we don't know exactly if they are, like Sarah said before, benign or pathogenic, and are called these variants of uncertain significance. I want to share with you the experience in our program at Institut Catalan of Oncology the last 5 years. I only picture here the most common cancer syndromes that are hereditary breast and ovarian cancer and colorectal polyposis or Lynch syndrome. Overall, the mutation detection is about 11%. In these cases, it's good because we know why they have cancer in their family, and we can perform cascade testing, and we can offer different surgeries, precision medicine, prenatal testing, and so on. But as you see, most of them are not solved in using the general panels that the Catalan government determined. So these families are the ones that were moved to this project called IMPACT. For those of you that have not heard about it before, this IMPACT project was created in 2020, and it was the aim of the project was to implement new tools for precision medicine that were not covered currently in the different communities. There are 3 branches, we can say, the predictive medicine or the core that we have heard about before, the data program, and the genomic medicine. The genomic medicine is led by Ángel Carracedo and is where hereditary cancer fits. Impact Genomica have developed, since its creation, a network of more than 100 hospitals with more than 300 professionals, and have representatives in all the autonomous communities of Spain. The main cases analyzed there is rare disorders, but also we have our pocket in cancer that I will share with you. All the sequencing have been done in 3 different sites, one being CNAG, the other Navarra, and the other Santiago. The work package 4 of Impact Genomica is the Cancer Work Package. It's led by Dr. Gabriel Capellà and has 2 parts, the hereditary cancer and the cancer of unknown primary. These are the steering committees of those lines, of both lines, and here is the management committee. How we work, or have we worked in Impact? Because Impact already finished last year. We established a network of clinical representatives in each autonomous community that talk with the physicians and select the unresolved cases of hereditary cancers that want to enter in this program. After fulfilling the accession conditions, the samples are centralized in 4 pathology departments in Spain, and they are sent to the 3 nodes I told you before. Then for hereditary cancer, all the results are first studied by our interpreter that is here in the audience, and is Mireia Ramos. And after her first interpretation, the cases moved to the interpretation committee and then to the expert committee. With all the results integrated, the report went back to the initial physician. Which omics we have applied? We have studied in most of the cases 3 to 4 patients, the germline, and from the probe and or index cases, we also sequenced the tumor. From the blood, we always analyze whole genome sequencing 34. And for the tumor of the program, we analyze DNA, but exome sequencing because it was less challenged than whole genome, and RNA-seq. It's a pity that RNA from blood was not included as Sergi thought in his talk, but it's something that is going to be addressed in the new Impact 2, because we believe that we are going to be more successful if we enter this RNA data. In total, in the different communities, more than 300 cases were presented and pre-selected. But at the end, only 168 were included for several reasons, because they were not fulfilling the clinical criteria, because participants at the end refused to participate, lack of material, or in a couple of cases because paternity was doubtful. This is the pie of the cases. Most of the cases are aggregation cases. That means that have 2, 3, or even 4 affected individuals, and a small proportion are unique cases, meaning that normally are very young patients without a prior history of hereditary of cancer. And this is the representation through all the communities, that it's strictly proportional to the sample size of the community, to the population. The pathologies that were collected, 57% were breast and ovarian cancer patients, 21% correspond to the gastrointestinal, and about the last quarter correspond to a miscellany of different cancer syndromes. This represents clearly the same proportion that we see in our genetic counseling units. Most of the cancer predisposition patients are from hereditary breast and ovarian cancer, and followed by colorectal or other GI tumors. This is a bit busy slide. I will not enter, and it's very technical, but just to let you know that what we decided is Impact was devoted to return clinical useful results. So we have sequenced whole genomes. We only analyze in the first Impact analysis a set of 105 genes, that were selected between all the community as the most clinical utility genes and that are clinical validated. So first we perform a single nucleotide SNV and short indels analysis, then CNVs and structural variants, and also deep intronic variants. Then when we have candidate variants, if we have the tumor, because in all the families we don't have the tumor, but if we have the tumor, we look in the tumor if we can see a second hit or a LOH or a specific signature that agree with the germline mutation that we think that can be responsible for this case. And the RNA of the tumor has been not used for the first step. We will use now in the second project. After we have all this done, the expert committee write the clinical recommendation, and the non-informative case moved to the Impact Bootcamp that I will explain later. In total, we have identified more than 2,000 variants, more than 700 were unique and were classified, and on average, in these 105 genes, we identify an average 4 variants. The representation of the variants according to their classification is here. a small number of pathogenic or lightly pathogenic, benign and lightly benign, and a great portion of the pie are BOSs. BSD in Spanish is a mistake. All this information are stored in the IMPACT platform, where we have all the results of the analysis, as well as a brief summary and also the reports. Here in the audience, there is some people that work very hard in doing all this work. At the end of IMPACT project, we were able to identify real pathogenic mutations that explain the clinical phenotype that we observed in the family in 10 cases, so 6% of the patients studied. It seems low to me, but compared to Sergi, it's higher, so not bad. Yes, I know. I'm joking. Most of them, some of them were reanalysis because they have been all lost or missed in the previous study, and some of them were mutations that were not, the technology used in the region was not able to identify this variant. For this, this slide, I use this slide to change to the next project, that is the IMPACT Buscan. This project was conceived in summer 2022 when the PMP program was launched for the government of Spain, and in only 3-4 weeks we have to build a project. It was like pretty stressful, but a group of people that were from IMPACT and were having all these challenges, we thought that it was better to try to study depth these cases. So the main points that we wanted to address is to use all the information that we have generated in IMPACT Genomica. We sequence whole genomes, but we only have studied 105 genes. And then we know, because we all are experts in hereditary cancer, that the number of variants will be very high, because if only in 100 genes we identify 4 or 3 for patient, in the whole genome will be high. So for this, we conceived the project as a platform for try to study variants of unknown significance from the genome data. And first, we have to identify which BOS we are going to study. That is one of the main parts of the project, as I will share with you. This is the final composition of the project. It's 9 centers in Spain, and the project is led by Meme Robledo at CENIO and by myself at ICO-IDB. Currently, there is more than 50 researchers involved in this project. This project is ongoing still, but I would say that more than 100 participate, because through different impact committees, they are also involved in this project. So as I told you, the main goal of our project is to coordinate a functional genomic platform to classify BOSs in hereditary cancers. For this, we define 4 specific objectives. The first we wanted is to enlarge our cohort, to increase the number of cases, because I told you that it was only 168, the budget that we have in the first IMPACT Genomica, so we decided to apply for a budget for 150 more. The second was to develop different algorithms to prioritize the variants, because we were aware that using the whole genome sequence, the number of variants will be huge. Then the main reason of the project, that is to functional validate the variants that we were identified. And at the end, and I can tell you that we have not arrived at the end, but the idea is to develop AI tools in order to help us to make decisions in the classification and in the clinical follow-up of these patients. This is the backbone of our project. So we came from IMPACT Genomica, and we have this core of families. I said that 10 were already explained, so we have 158 really to move to here, to the work package 3 of the project, that is bioinformatics, followed by the variant selection committee, and then work package 4, that is functional validation. And I want to enter in all of these objectives very briefly. For the first objective, we have already selected the 150 patients. The pie is similar, although there is a bit more of proportion of single cases. And per pathology is very, very similar to that square that I show you, breast and ovarian and digestive, and then the others. Work package 3 and 5 is led by Dr. Fatima Al-Sharour at CNIO and by David Gonzalez Cabrero and Navarra Biomed and by their teams. The idea that they have been working is to digitalize all the information and the pedigrees, to collect the NGS data from the different sites, and to perform some algorithms that give a list of variants that the variant selection committee can play with. In this slide is very summarized. I don't have the time for the details, and it's not my expertise neither. But for single nucleotide variants, we have Dido Carrero at the CNIO, at the group of Fatima, that have developed a score, the DD score, we call Dido score because it's her name, that range from 0 to 1, and they integrate several prediction programs, data from several prediction programs, and also from databases and from the literature to create a list of variants that can be prioritized with the DD score, apart from other information. In Navarra Biomed, Edurne Urruti and Estefania Huergo, with David, perform the pipeline for CMVs and structural variants identification, and they provide us an Excel form. And we are very lucky that at the last part of our year, Gerard Muñoz joined the Dr. Surrayes group. That is one of the groups of our team, and it's expert in RNA, and it's providing us some light in the RNA from the tumors that is not easy at all. So after we have this variant list, we move to the committees of selection. We have 4 committees. The breast and ovarian is led by Ana Vega in Santiago and Miguel de la Hoya Clínico San Carlos, Alberto Gascón at CNIO, the endocrine tumor, Clara Ruiz in Santiago, the digestive tumor, and Barbara Rivera in our center at Titibel, the other tumors groups. And this is the person that work with the different groups. So they work with the prioritization reports that are provided by CNIO and by Navarra Biomed, with all the information from the past project of the 105 first genes analyzed, and they have access to the different GPAP platform in the 3 sequencing nodes to visualize all the data that they want to review or want to see. With this, they apply different filters according to the intrinsic nature of this pathology, and then they generate a list of variants. We also generated a list of variants of our cohort at the same time. And they move to the work package 4. Work package 4 is led by Laura Valle, also in our institute in the debate, and Cristina Montero at CNIO. Before they have the first variants to arrive, they have worked with all the groups in generated a map of resources, a map of gene experts, of pathways and methodologies. That way, by this means, when we have a variant, they easily can identify which group would validate better and which needs what they will need from the families or patients. The first track, we have a fast track list to move contains the variants that clearly we think that have to be validated. Normally are, as you can see, very deep intronic variants or structural variants, because are in genes that are clearly responsible for the phenotype of the patient and probably were missed in the laboratory of origin. We have this list of variants, and we have the center and the person who is responsible, and if they finish the final classification. to start to wrap up, I think that if it's okay, I will present to you the first case that was resolved in our project, in the Impact Bootcamp project. It was done here in our group, in EDBA, by Paula Rofoes, a postdoc in my group. And it's a deep intronic variant in the APC gene. In fact, I will present only one, but this type of variants appear in two families at the same time when we were analyzing. These families both have a clear polyposis phenotype, and the centers of origin didn't identify any mutation in the genes related with polyposis with the standard clinical methodology. And in both cases, we identified a mutation that, as you could see by the number, that is very deep inside of an intron. I want to explain for time constraints the first one. The first one was one family was from Madrid, that is this one, and the other was from Zaragoza. This family has a deep intronic variant deep inside of an intron that is shared by the three affected patients of the family, two sisters and a son of this woman. When we apply different splicing prediction tools, Splice AI highlight that there is a gain of an acceptor and a donor splice site. Reviewing in the literature, we realized that variants in this region, but not the same variant, were published previously by the group of Stefan Arets in Bonn, and both of them that are located here and here are generating the same, the insertion of a cryptic exon or a pseudoexon that was out of frame and producing a truncated transcript and protein between exon 4 and 5 of the APC gene. Our variant is here, is not the same, and our experiment was to try to see if RNA study of this variant in the affected individuals produce an abnormal splicing. So we designed different PCR assays following the Enigma and Clean Gene recommendations for RNA studies, and we were able to detect an abnormal band in the three affected patients. When we sequenced this abnormal band, it was exactly the same effect that the other two variants were showing, that is the insertion of this cryptic exon. So with all these results, we proceed to classify the variant according to the ACMG guidelines. And we were able to give points for the effect that we saw in the RNA, for not being present in population database, and also for the specific phenotype that this family has. Altogether, if we apply the rules, this variant has 6 points and is moved to likely pathogenic variant. With this result, we write a new report with this data. We talk to the clinician, and then these clinicians can use this information to handle with this family and other relatives of the same family. And it happens the same with the previous family I showed you from Zaragoza. So just to finish, 2 slides. One, what we have learned of these 2 big projects. And we have developed a lot of protocols and guidelines. We have signed a lot of DTAs between centers. It has been a titanic work, especially administrative work, to move this project. But another thing that we are proud to construct is that we have created a national network of different experts. We have an interpretation committee, an expert committee, we have a group working with the sequencing nodes. So we have really, I think, tried to provide equity in Spain, because all these resources were needed, because when we received the data from the different communities, autonomous communities, it was very different. So I think that we have done a big effort to coordinate and to do the same. There is strengths that are the ones that I told you, and there is also weakness. I will say that maybe the short duration of the project, because it took us 1.5 years to start Buscan, just signing DTAs between centers. So everything is very slow because it's the whole Spain, 70 communities, and it was difficult. Some of the families were having complete clinical data. The circuits at the beginning were slow in all the cases, sending the sample, the pathology, everything. I mean, it was very difficult. But now it is here. And the other thing is that there is very little budget for human resources. So I would say that here I can see 15 people that is not paid by Buscan and have invested every week more than 6, 7, 8 hours working in this project. So this is one of the weaknesses, but hopefully in the future somehow can be used in the national health systems for good. I want to finish to thank all the participants. Most of them are in these pictures, but not all. The Buscan team in the first retreat we have just a month ago, and the IPAN Genomica in the Carlos III annual meeting. But in the last minute, I want to share with you a video that we have recorded for the first family and for the first clinical oncology that have this family. Because I don't know, but I think that for me, one of the most rewarding parts of this project, and I think that for most of our team is like this, is to think that behind every experiment, behind every clinical decision, every committee, there is a patient, a family, a human being that the work that we do really matters for them. And you will see here. I really hope you like it.
Para saber todo eso y generar el problema, porque realmente no lo sabíamos. Para nosotros yo creo que esto ha marcado un antes y un después, porque el saber quién es portador y quién no cambia completamente la manera de enfocar los controles y las decisiones médicas. Y además en mi caso, que actualmente estoy embarazada, pues pensar que con un simple análisis podré saber si mi bebé es portador o no, pues eso da muchísima tranquilidad. Lo más importante, yo creo que de cara al futuro, es podremos romper el ciclo de la enfermedad y tener hijos libres de este gen. Entonces es una sensación de alivio total.
Un agradecimiento que personalmente yo siento a toda la gente del laboratorio que estaba detrás peleando ese resultado hasta llegarse acá.
Y también, pues, de alguna forma resaltar cómo, pues, trabajando en equipo se logran resultados como este, y eso también es muy importante y nos da una experiencia muy positiva en el trabajo en equipo también a todos nosotros. Muchas gracias. Yo solo diría que muchísimas gracias a toda la gente que ha estado involucrada en el proyecto y que la verdad que estamos contentísimos, es lo único que puedo decir. Les voy a dar también las gracias a ellos de parte de toda familia por habernos propuesto y pues eso, sobran las palabras.
Thank you. Thank you. Thank you. I think it's nice to see that there is a patient, no, on the other side of our life. Sarah.
Thank you very much. Indeed, it's a big effort from many people involved. Congratulations. So I am involved in some interpretation committee, so I know from inside the work that it implied. So I was thinking, after these projects that illustrate, and we are learning, that implementing these standardized, homogenized genomic techniques, do you think, or are you in the experts in the direction of the projects envisage how to change the clinical practice, implementing the learning, that the knowledge that we have got with the impact boost, impact 2?
I don't have a clear answer, but what I think is that health is transferred from the Spanish government to the different regions, and this is a challenge, no? I think that there is some meetings that try to not to take advantage of all that impact has generated, not only for cancer, but also for rare disorders, and try to bring to the every health conselleria, or how it's that, in every community. And I think that if the different communities have the interest of working together, I think that there can be mechanisms to do it. And with all the works, all the standard procedures, and all the things that we have done, I hope that they are going to use, but I don't know. The answer is I don't know. I hope so. Okay, thank you. Thanks so much. Can I have some?
So it's my pleasure to introduce Roberto Pinheiro, and well, I'll hand it over. This moves a little bit again.
So hello everyone, and thank you, CNAG, of course, for bringing me today here, because I'm a newcomer, let's say, in the blog. It's my first project in collaboration with CNAG. So we're going to switch gears a little bit, because we come from a large effort, and this is a much smaller effort, but still, I will try to show you some preliminary data that we're getting in this project, in which we are interested in understanding the biology of these cells that are the so-called circulating tumor cells. And I like to start because, as I said, it's my first project involving genomics, particularly at the single cell level. So I was trying to find something to bring me up, cheer me up. So I find this quote from Edmund Beecher, who wrote in 1896 this book, The Cell in Development and Inheritance, and he wrote he quoted this sentence: "The key to every biological problem must ultimately be sought in the cell, because every living organism is, or at some point, as being a cell." And I think we are all agree that this is today very relevant, particularly in the field of cancer genomics. So I'm not going to spend much time in what is the etiology of breast cancer or the figures, because I think we are all aware of it. But we know that is the main leading cause of cancer, or the main diagnosed cancer in women worldwide. And also because it has a quite large impact on what is cancer-related deaths. In this, if you take the cancer statistics from USA, it's the second leading cause. But if we look at the Spanish Society of Medical Oncology numbers, in Spain is the first cancer-related death in human population. But we know that we have made a lot of progress during the last years in terms of diagnosis and treatment, but still, roughly from 5 to 10% of the patients, when are diagnosed with breast cancer, they are already diagnosed with advanced stage, with metastatic breast cancer, right? And the truth is that those who are not detected at the beginning as metastatic breast cancer, 30% of those patients end up becoming or having a spread disease. And that's the real problem in this disease, because metastatic breast cancer, as many metastatic diseases, is an incurable disease. And that's why we're trying to understand a little bit what's happening in this process, right? And I'm bringing this slide because I don't going to go into detail of what is the complexity of the metastatic cascade, because I think everybody is quite aware of this. But just to remind you that during the evolution of the disease, from the primary tumor or even from metastatic lesions, there are some small populations or small number of cells that are able to detach from the lesion, from the primary tumor or the metastatic lesion, invade through the tissue, and infiltrate the circulation. The blood vessels are going to the circulation, and that's what they call the so-called circulating tumor cells. There's a lot of literature in the last 10, 15 years in this field, but we know that these cells, thanks to the development of the liquid biopsy technologies that allow us to sample a body fluid, in this case mainly the blood, and search for these cells in the blood of the patients. And we now know that these cells can be found either as single cells, as you see here, or as a small cluster of cells, right? Problem being is, as I said, low frequency of these cells. So we need to screen the blood and then stain those cells for specific markers in order to at least tell with some sort of level of confidence that we are talking about cancer cells. The ultimate way of knowing it is just doing some genomic analysis to see aberrations. But as I also would like to mention that this small cluster of cells, sometimes, or even the single cells, are helped by the microenvironment, as you can see here. So you see a CD45 a leukocyte marker positive cell that is attached to 2 cancer cells, or another one here that is attached to 1 cancer cell. And we know that that's important because not of my work, but work of previous in literature, they show that these cells are able to modify, for instance, the proliferative profile of cancer cells and help them into the metastatic process. So, and we believe that, understanding the biology of these cells, being able to sampling these cells, analyzing it, will allow us to in a way, to know better those populations that are the metastatic competent cells, or the mix, right? But as I said, this comes with a problem, and the problem is the numbers. If you take a 7.5 milliliters, so ideally tube of blood, and you start screening for CTCs, what you end up is a picture similar to this. These are images taken with the gold standard in the field, that is the cell search assay, that identifies CTCs based on the expression on EpCAM, the surface protein, and then stain for cytokeratin. And so everything that has been collected in here based on EpCAM and express cytokeratin and do not express CD45 is meant to be epithelial cells in circulation, is meant to be a cancer cell. But they come in different sort of numbers and flavors, as you can see in this picture, right? If you look at here at the enrichment, you might end up seeing 1 CTC white blood cell cluster in millions of white blood cells, or in this decade or so, most of them coming from this group, from Nicolas Aceto's group, first MIT and now at Zurich. In different papers, he showed that these clusters, they have a much more metastatic potential than the single CTCs for a variety of reasons, because are linked sometimes because they are epithelial cells that still have some young-to-young, cell-to-cell unions that allow you to survive better in the circulation, because they are surrounded sometimes by platelets that allow you to protect from the immune environment. So there are few already. We know some of the reasons why these clusters or these CTCs might end up being the ones producing metastases. But interestingly, some papers showing that they might have oligoclonal or polyclonal origin, meaning different cells from different clones cooperating in order to succeed in the process of seeding metastases. So basically, this is something that, in a way, define the paradigm that we initially had, that is 1 clone, 1 metastasis, right? That's when I got kind of interested in trying to understand the biology of these cells, linked to the fact that some genomic studies have shown the existence of, in breast cancer, metastatic breast cancer tissues, the existence of multiclonal metastases. But to put in perspective this work that we're trying to put forward, here I try to summarize most of pretty much all the genomic studies in which either single cell or CTC clusters has been profiled, right? As you can see, not many are there. Particularly if you compare the ones that are analyzing single CTCs and the ones that are analyzing CTC clusters. For you to see it better, I just highlighted here, for instance, this paper by Jackie Shaw group in 2000, I think it's 14, that they did targeted PCR for an exome in TP53, and they compared in single cell and pool cells, but also CTC clusters. So interesting, but we are lacking a lot of information in that case. So that's why we need larger studies. There is this other study that what it does is to do low-pass sequencing and copy number profiling of 53 clusters isolated from, in this case, I think it was particularly in early breast cancer patients, because they are less frequent in early stages, but we can find it there too. And then the most recent one, again, coming out from Nicolas Acetor's group, is this one when they do whole exome sequencing of cluster isolated from 7 breast cancer patients and 2 prostate cancer patients. And in this paper, hard to understand for me because there's a lot of bioinformatics involved in this in order how to do clonal deconvolution and things like this. But they found evidences of the presence of polyclonal clusters in the blood of papers, right? That could link to what his more basic research tells us. So that's where we come in. This is our sort of the design of our study. We are trying to compare single CTCs in CTC clusters and also with tissues when available. This is the characteristics of the depth of the, let's say, the sequencing data we use for single CTCs, for clusters, and from bulk DNA. So basically, the rationale of this project was, okay, it's put forward that the CTC clusters are heterogeneous and might be responsible for polyclonal metastases. So we are not aiming that high, but at least we wanted to kind of shed some light on what is the heterogeneity or the complexity, the mutational landscape of these 2 compartments that are running in the blood of cancer patients. This work has been mainly done by this person here, Cristobal Fernandez Santiago, a very talented PhD who spends half of the lab doing wet lab and the other half doing bioinformatics training. And is the one that's been in charge of most of the sample prep, but also in the bioinformatic analysis with the help of David Posada and Joao Fernandez Alves from the University of University of Vigo, and also some collaborations in the University of Düsseldorf, Hans Niebauer and Andre Franket, but also the collaboration of CNAG in the analysis on the whole exome sequencing of these DNAs. So basically, the workflow is in principle quite simple. We have patients diagnosed. In this case, we focus in luminal breast cancer in order to have a sort of homogeneous cohort, and also because it's the largest proportion among the clinical subtypes or the molecular subtypes of breast cancer. So we went for luminal breast cancer. And what we tried to do is when those patients were diagnosed with metastatic breast cancer, we took a liquid biopsy. And we're focusing at a particular moment, and it was done in purpose. That is, those patients have been already subjected to different lines of treatment, because that is a very advanced disease. So the chances of finding these cells in the blood is much higher, and I'll show you later. So what we did is try to use the CellSearch platform. That platform captured the cells in this sort of cassettes in here, and then what we did is a very rudimentary approach, but is to put those in a slide under the microscope and fish them out by micromanipulation, right? And we try to end up with pairs, single CTCs and clusters from the same patient. That I tell you that is quite challenging. So then we took the FFP from primary tissues and metastatic tissues when available, and that's what's also subjected to whole exome sequencing. Well, first, single cell were subjected to whole genome amplification and then sequencing. And then what we're trying to do here is just a comparative or exploratory, let's say, analysis of what we found in these 2 compartments, but also in relationship to the primary tumors and the metastatic tumor. So just to spend a minute in the cohort, this is a small study, 128 patients, but it will become even much smaller, you will see later. But I only bring forward this because, as I said before, this is a polytreated cohort of patients in order to maximize the number of cells that we could get out of the blood. In a previous study, the one I showed you before, for instance, patients bearing 1 CTC cluster at the diagnosis, it was at 25% of the patients in a 64 total number of patients. So, and in this study, we went up to almost 37% of patients. So we are definitely, using this cohort that might not be, from the clinical standpoint, the best, but for the purpose of this project, I think it was better to use that. And as you see, CTC and CTC clusters, they tend to correlate with the prognosis, despite of being patients that have undergone at least 3 or more lines of hormonal or CDK inhibitors therapy. From the technical standpoint, so basically all those single cells or clusters that were isolated were subjected to whole genome amplification with this kit, the Ampli-1 whole genome amplification kit from Menarini Biosystems. That is basically a ligation-based PCR where the genome is just cut by restriction enzymes and then ligated to adapters and amplified. We choose this approach based on previous work done by David Posada in Vigo because, perhaps this, compared to other sorts of kits of whole genome amplification, has less bias or has less allelic dropout. And then what you do is a PCR just to QC your samples. That is basically is a PCR based on 4 fragments across the genome that you're looking for the genomic integrity. So this PCR gives you a GII, that is a so-called genome integrity index, where 3 or 4 numbers, so basically 3 or 4 bands in your PCR means a good integrity of the genome, so suitable for NGS analysis. And lower than 3 gives you a sample that probably better not put it in the sequencer because you might be wasting your money. So from the CTCs and clusters that we got, after whole genome amplification, we end up with an 85% of cells that we analyzed here, and a 77% of the clusters in here. So basically, compared to another project in which we did that, but in mouse models, we have a much better success in this case. And I said you before that the cohort becomes smaller because at the end of the day, you want to compare from the same patient single CTCs and CTC clusters plus the tissues. So we end up selecting these 7 patients. We have this patient here from which we have, from this person we have 3, let's say, follow-up samples at different time points of the disease that I showed you later. And then, but out of the 7 patients, from 4 of them, we have matched primary tumor metastasis, but also CTCs, single CTCs, and CTC clusters. So this is what it went into the CNAG sequencing machines. We did that, as I said, we tried to answer a priori very simple questions. For instance, what mutations are shared between single CTC cluster, or what one are private mutations, if any, for instance? Are the mutations common across patient or just specific of a patient? Whether one compartment has a more complex or less complex, let's say, genomic landscape than the other. Whether these CTCs, can you use it as a surrogate of the primary tumor or the metastatic tumor? Because at the end of the day, we're talking about metastatic breast cancer patients whose tumors has been resected probably many years ago. So we need to find out whether it's a proxy of the tissue, what we have in here. And then whether these CTCs or clusters come from the same clones in the tumor, right? That's a very interesting question for us to understand. Apologies if this is too very basic, but I need to understand, get to grasp. I'm a geneticist, so it's my first approach to this. But the first thing, discrete data I want to show you is that, we put here all patients at the sort of patient distribution of the mutations that we found in the CT. This is specifically CTCs, and it's altogether pooled CTCs and cluster. And we found that it's quite homogeneous what we find in terms of silent, non-silent mutations, tronic mutations across patients. But we find this patient here that is a kind of outlier that we need to address. Why is that? It might be related to the clinic. It might be related to the technical approach used in this project. Basic numbers. If you compare the number of mutations that we found in single CTCs or in CTC clusters, you see that they are not statistically different, although we tend to have a lower number of mutations, overall mutations, in the cluster population than in the single CTCs, perhaps unexpected, perhaps not. We don't know. Still, the main conclusion is these two populations at least are very homogeneous in terms of number of mutations that are bearing. Don't take this as a final data because we are still applying different filters and perhaps even changing the colors, because one thing I've been learning along the project is what sort of colors should be used when you're using for bulk mutation, for bulk analysis, and what for single CTC, particularly because cluster might be 2, 3, 4 cells. So, and you are using colors for single cell when you're using a small bulk, right? So something that we need to figure out, but trying to be more descriptive. So no difference in terms of what is the mutational burden in terms of numbers, but numbers perhaps don't tell you all the truth. And what's trying to do here is just to plot the distribution of the mutations based, or each point should be like a mutation and the percentage in which is present either in the single CTCs in the x-axis or CTC clusters in the y-axis. And of course, what we see is we find cells or mutations that are sort of more enriched. If we take an arbitrary cutoff of more than 50% of CTCs bearing this mutation, or more than 50 and less than 50% of clusters. So we have some mutations that are enriched in single CTCs, some other mutations that are enriched in CTC cluster, but we also have a number of mutations that seems to be exclusive or single CTC, and a number of mutations that will be exclusive in CTC clusters. Still, population that seems to be quite homogeneous, but perhaps the distribution of what those mutations mean should be relevant to understand the biology of these cells. And if you look at the per patient distribution, although we see a lot of some heterogeneity, what it seems to be the case is more cluster dominance. Those mutations that were present in more than the 50% of the cluster we analyzed, but less than the 50% of CTCs, are the ones making up the largest population of mutations found in most of patients, particularly relevant in this patient, in the 441, that is quite large, and even in this one. But still, you can see some sort of level of heterogeneity in there too. And again, this patient, the number 66, we have, again, in this case, also the distribution of mutations, so the pattern of mutations, how they distribute is different. But we ask ourselves, well, what do mutations do appear in multiple patients? So it's just that you find it in a CTC or all the CTCs of a patient. And it's when trying to represent here that, as probably expected, most of the mutations we find are, patient-specific. So we need to look at what sort of mutations, because importantly, something that I must acknowledge is that we don't have healthy control, okay? So we're using healthy panels in this case in trying to filter out what would be germline mutations. Still, that and also playing with the variant, the frequency of the variants to filter out those mutations, but we might have some contribution. That's why we are still playing with different colors in order to understand better this. But if you zoom in this, so this would be some sort of numbers of mutations that are repeated consistently found in 3 of the patients, or in 4 of the patients, 5, 6, or 7. So with that, a small number of mutations in here that seem to be common to all patients, but we don't know what the mutations are, right, yet. So if we look at the distribution in terms of whether I come from the single CTCs or from the clusters, most of that, as you can see, for instance, mutations that we found in the 7 common to 7 patients, they are found in both populations, in single CTCs and in clusters, right? And very small proportion of mutations that might be specific or only found in clusters and only seen on single CTCs. But we are trying to understand also how, as I told you before, how this mutation reflects what is the mutational burden of the tissues, of the primary or the metastatic. So what I'm trying to plot here is just the Jacquard index that tells you a sort of measure of how homogeneous or how much these compartments overlap. And again, it looks like what we found is a quite homogeneous picture, to be honest. You have here the single versus the primary tumor, the single versus the metastatic tumor in the patient that we have that, the cluster versus the primary tumor, and the cluster versus the metastasis. And as you see, the numbers quite sort of repeat themselves quite frequently. The only thing we can see is in some cases you see that the cluster number is slightly bigger than to the single in relation to the metastatic tissues. That might tell us something. We don't know yet. This sort of a kind of different analysis in which I'm plotting here the number of mutations that cluster of single CTC share with the metastases, with the primary tumor, or with both compartments at the same time. So as you see, the total number is probably related to the lower number of mutations found in CTC clusters. We also have less mutations sort of shared between cluster and metastatic primary tumors. But when we calculate the Fisher test and we calculate odds ratios in how likely are the mutations of single CTCs or how likely are the mutations of clusters to be shared with the primary tumor or the metastasis, the odds ratios we found here. So in terms of the metastasis, there is a small sort of, the CTCs, single CTCs tend to share more mutations with the metastasis. That to me perhaps was a kind of a surprise. I don't know. We need to look into this in deeper. But CTC cluster had a larger odds ratio, meaning a larger 38% of the mutations in clusters could be shared with the primary tumor, right? What would be the meaning? Probably, it could be related to what clones are shedding these clusters from the tumors, whether the single CTC are more diverse, more heterogeneous, while the clusters are more conserved. And that is something that we're trying to understand too. And one approach we're doing that is just to trying to, we're using the cell5 pipeline that David Posada developed, published like a few years ago. And we're applying those sets of mutations that we find for each individual patient and try to create these phylogenetic trees to see what is, the relationship between cluster singles towards the tissues. And what you have here is the primary tissue for this patient and from this patient, the metastasis. And something that calls our attention is across the 7 patients we have, this is a sort of reproducible picture in most of them. And it's the fact that the clusters tend to cluster in a different branch are related to the single CTCs. As you see, the single CTCs tend to be more scattered in different branches, but the clusters seem to have sort of a common ancestor and closer to the tissues. And this is also something that you can see in the length of the branches. So the larger is the branch, the more evolved or more divergent from the ancestor clones is the CTCs. So that could be something interesting if it holds true, okay? If it holds true, it would be interesting because it might tell you that the clusters are better at resembling what you have in the primary tumor or in the tissues at least. And just to end up in the last few minutes, I'm not sure how I'm doing on time, but I showed you that they have this patient with follow-up sample data. When it was diagnosed, this patient underwent the treatment, and we were able to, or we managed to get samples at initiation of the fourth-line therapy with ADC, antibody-drug conjugated sacituzumab govitecan, the anti-TROP2 targeting TROP2, and then during the evaluation of this treatment and at the progression of the disease. And this was a very much striking patient because, as I told you before, the numbers are huge. This is very rare to see in a patient this number of CTC. At this point in time, this patient had more than 2,000 CTCs in one blood count in 7.5 milliliters. That's a lot, and 66 clusters. That's a huge amount of clusters in there. So you can imagine, what patient is this? She initiated the therapy, the antibody-drug conjugated, and at the beginning it sort of worked. And I'm saying it sort of worked because, still many of the CTCs disappear. So probably meaning the therapy is working at reducing those metastatic lesions that are releasing the CTCs, but still very high above of the 5 CTC count that I told you before. That is the threshold for good prognosis. Interestingly, clusters disappear from the blood or were not identified by this technology, so search assay. And at the time of progression, numbers of CTCs went up and clusters show up again. And we were lucky to be able to fish some of them out. So we ended up sequencing 9 single CTCs, 5 clusters from the same first visit, 14 single CTCs here, no cluster, of course, to sequence, and 9 and 4 clusters out of the 5 with there in there. So I'm just showing here the mutations that we found across the 3 visits. So the CTCs always vary in these mutations. The large proportion of that, that's no surprise. But then we have a small proportion of mutations that were only observed at one time point. And I'll try to illustrate it here in this sort of basic heat map. When in sort of pink, you have those mutations that were always found in the CTCs, so in the three visits. But we have some sort of mutations that were just only found at the beginning of the treatment, that with the treatment then, we don't know with the treatment whether it's cause of the treatment, but after therapy, at least disappear. Okay, we cannot do a causal effect that is because of the treatment, but at least we cannot detect it, at least in the samples that we're analyzing in here. So these are interesting mutations because, as you see, at the time of progression, there are only 2 or 3, but new mutations showing up. That will be very interesting. So just to show you that somehow the CTCs are able to reflect the dynamics. And the interesting bit was also when we do the phylogenetic analysis in all these samples that we got. Remember, at the first time of the visit, as you see the clusters, again, small branches, and tend to be closer to each other, unless this one over here. However, single CTCs were more scattered across the branches. Again, looking probably at more clonal mutations, perhaps, or less evolved as compared to what we found in the CTCs. If you look at the BC2, when the numbers went down, you have a scattered number of CTCs in here, but if you look at the position of the branches, they are more closer to the tissues, to the primary tumor and the metastatic tissues. So perhaps, the therapy somehow is selecting those core mutations in this tumor. And in the third visit, you have some cluster up there and cluster here, and then you see probably, and that's pure speculation, due to the selective pressure of the therapy, those single CTCs and long branches emerging in there. So affecting, doing some subclonal selection. Of course, this is not very much a clinical tool. We understand that. But potentially there's something in a view how we can envision if we're able to find some interesting core mutations that will be, how can we address whether they're clinical relevant? The idea will go to an approach will be to look for mutations that are low buff in the primary tumor, but mid to high buff in the metastatic that we find also in a relative or relevant proportion of CTCs, and design some digital PCR assays to look in all the cohort of patients in the plasma of the patient in the disease-free DNA and whether we can relate it to the prognosis, right? That will be a way forward to try to bring, perhaps relevant mutations in the content, try to follow up the disease in these patients. There's no take-home messages because, as I said, this is quite preliminary, but we can anyway so far conclude that these are two sort of homogeneous populations, although there is a small level of heterogeneity, maybe how these mutations are distributed. Most of there is a predominance of shared mutation by single CTC and CTC cluster across patients. Recurrent shared mutations tend to be core mutations present in both, as I said. The vast majority of primary tumor and metastatic mutations are present in both. The important thing of the phylogenetic analysis is the fact that it might be that the single CTC or the clusters seem to come from a same region of the tumor, close, in terms of clone, while the single CTC, they be much more divergent. And we observe some sort of changes in the clonal composition of the cells along the disease. And with that, I would just to thank the people working, the clinical team led by Rafael Lopez in Clinical University Hospital in Santiago, my small lab, and people from Oncomed who collaborated with the group I belong to in Santiago, our collaborators David Posada, Joao Ainair, Tania Feng, Hans Niebuhr, and Franken, and the people supporting us in the CNAG with analysis and the funding entities. And with that, thank you for your attention. Was it too long?
I don't know. Questions? Yes, please.
Thank you very much. So I have a question about the mechanistical part and looking at RNA. Have you considered it?
Excuse me, sorry.
So for looking kind of, adding the next layer to reveal the mechanistical part, have you considered looking at the RNA? What differentiates kind of the individuals versus the clusters? And how do they behave when they are together? So what changes there?
That's my goal, actually, to have the same readout either from the same cells or at least from the same patient to have DNA and transcriptomic data. It's something that we haven't done so far. We have done either single-cell DNA, and I'm trying to move forward into transcriptomic. I'm doing some transcriptomic, but just looking at what is publicly available data, particularly from Nicolas Aceto group that they have published a lot, and comparing the transcriptome from single CTC and CTC cluster, and we find different things, right? Even we have, it's not published yet, but we have derived a signature that is genes upregulated in CTC clusters compared to single CTCs, that when we look at those genes at the primary tumor level, we can sort of predict distant metastatic-free survival events. So, but that's something that definitely, the problem with the transcriptomic and the genomic is screening for these cells, you have to stain them. So the processing of the sample, you don't know when the sample is coming through, whether at 9 a.m. or at 2 p.m. You know what I mean? So doing all in the same pipeline, so fixing the cells is quite a helpful way forward of doing that. Yeah, but we're working on that. Thank you.
Hi, I would like to ask, you spoke about the clear correlation between the number of CTCs or whether they are clusters and the better or worse prognosis. I wanted to ask if, do you plan on looking for any correlation between the number of CTCs or the mutations that they carry and the sites of metastases? Or
That's a very interesting point, because at the end of the day, we don't know where these cells come from. So you found it in the blood, but some of these patients have perhaps liver, lung metastasis at the same time, so you don't know whether they're coming from one site or from another. So the idea here, I was perhaps a bit naive at the beginning and say, okay, let's see what are those core mutations found in CTC cluster, and then go to the tissues and then try to digest to see whether we are able to pinpoint where they're coming from. That, and I think that now coming to terms with the development of this project, I understand that that's going to be very difficult, but still, it will be interesting to find if some, perhaps, sublime mutations that they're not driving of the disease, but they are needed to the maintenance and the progression of the disease. As we see that, that's a mutation that don't go away with therapies, right? So
I was thinking a bit more on the previous step, so whether CTCs coming from the primary tumor could help you know where the metastases could go. So think about the CTCs finding a niche in other tissues. So if they carry specific mutations, that they will give them advantage to metastasize in the brain or in other tissues.
It's a good point. I would perhaps focus more on methylation status, because with methylation you probably can pinpoint.
But even that, because it's going to carry the methylation from
the primary tumor. Yeah, it's true.
But it would be a neat one to try to figure out. Mm. But you could take attack sequence, the single cell attack, that is sort of a good reference for an epigenetic fingerprint.
Thank you.
Good think about it. Yeah, thank you for this.
But it's difficult.
Yeah. Thank you for this question.
Okay, if there are no more questions, let me go to Christophe Fleury from Oxford Nanopore. J'ai 15 minutes.
Je suis déjà en retard. All right. Good afternoon, everyone. Happy to see there are still some people. Don't worry, I'm the last one, so we'll try to do it in 15 minutes. So my name is Christophe Fleury, and I'm a clinical cell specialist at Oxford Nanopore. And I'm here today with my colleagues, Alfonso Troveno and Luisa Vicevani, and also Nahuel Manzanaro. And first, I want to ask everyone, so that's going to keep you awake, who does not know Oxford Nanopore? Everyone knows Oxford Nanopore. Great, so I don't need to tell you that we're doing sequencing, right? We're a sequencing platform, and here at CNAG you have access to this Promethean, the big instrument here. There's one, and there are two, I think, on their way, at least. Yeah, it's two. Yeah. And the topic today was about cancer, and we've covered a lot of ground, from research all the way to diagnostic and treatment of cancer. And as you know, before reaching that point on the right-hand side, you need to start from the left-hand side, right? And the discoveries are going to feed the chain there to come all the way to the right. And Oxford Nanopore is on this odyssey from the left-hand side to the right. And I was looking at the program when I prepared this presentation. I wanted to try to cover a lot of the presentations and the topics that were covered. So first, we're talking about cancer, and you recognize on the left here the 8 hallmarks of cancer and the 2 enabling characteristics from Hahn and Weinberg, right? And to study these, you need to look at an awful lot of different mutation types. SNV, structural variants, we've heard a lot about structural variants today, epigenetics as well. And you need to look at the microenvironment, not only at the bulk level, but also at the single cell level. And the message here is that whatever you need from this, Nanopore can provide it to you. And we have, either through our technology, funding technology, or through partnerships with 10x for single cell, we have solutions for you. But I wanted first to look at the transcriptome. You've heard today about RNA-seq, and usually it is done with short-reads and RNA-seq. Now, one thing that can be really difficult with short-read RNA-seq is to have a good idea, a good mapping of gene fusions, like in leukemia. It's difficult to quantify, if not impossible, to quantify exactly the isoforms. And this is something you can do using Oxford Nanopore. We have kits that enable you to do this. We have single cell, of course, and a spatial transcriptomic with Visium from 10x that we're going to talk on the next slide. And one thing that is quite unique to Oxford Nanopore is to look at the transcriptome in its native form. Do you know you can look at the RNA without doing a reverse transcription and amplifying it? In this way, you can capture not only the isoforms and quantify them, but also look at the RNA modifications. We don't know if this has a role in the translation into proteins. We don't know its role. But that's something you can look at with Oxford Nanopore. Then I was talking about Visium and CNAG Genomics. We've heard this morning a presentation that showed this Visium HD, that is a very high resolution spatial way to look at the transcriptome and the tumor microenvironment. You can do, with short reads, gene level cluster analysis of your cells. But are you not missing something? Are you not missing the isoforms that will reveal something about the cell functions? You'll be able to look at the exact proteins that might come up. Ideally, you want to do single cell proteins, but we're not there yet. So with looking at long reads from on platforms like Visium or the Chromium technology, you'll be able to have these clusters that are based on the transcriptome, so 60 to 100,000 molecules instead of 20,000, and you'll be able to map your SNPs, your isoforms on this pretty Disney plot or UMAP projection. And there are already publications in the leukemia field in particular. And if you're interested, please contact us and we can give you more information. Now, if you go back to the DNA, one thing that you may know about Nanopore is that we can study the DNA in its native form. And we're going to have access to not only the SNPs, the small variants that you can detect perfectly well with other technologies, but also to larger, more complex variations like gene translocations, CNVs, etc. You're going to have a look, you're going to be able to look at the methylation without any enzymatic or other chemical transformation. You're going to directly look at the modifications. So not only 5-methyl-C, but 5-hydroxymethyl-C or 6-methyl-A. Technically, you should be able to look at all the others. We just don't have all the models. And you will be able to look at the dark genome. This is something we don't talk too much about, but if you look at the short-read whole genome, you're missing 6 to 8% of the information. So looking at whole genome and what it can bring to cancer, and here this is the only slide, I think, about the workflow, because every time when you look at the DNA, we're going to have one workflow, including when we do targeted. And here you see on the top, you can take a sample from blood and look at the, do a whole genome 30x and sequence on the promethazine and do analyze with our tools or other tools. I mean, a lot of the tools are on GitHub, and it's really fun for the bioinformaticians. I know there are a lot in the room, but the Nanopore data give and long-read data in general is still something that needs maturing, and therefore bioinformaticians have maybe more fun than with conventional data, or maybe problems, but I hope you have fun, not problems. If you want to look at somatic variants, right now, unfortunately, there's not a somatic-only way of looking at it, right? You will need to have your tumor sample and your normal sample and do the subtraction of the germline variants. We're getting there when it comes to somatic only. There's just one restriction, I have to admit it exists. It's not going to work very well with FFP samples. You have to have fresh samples. The type of data that you get from a Nanopore sequencing is, as you can see there, very interesting, because not only you have the long reads, not only you have the methylation, but you're going to also have the allelic resolution of your variants. Meaning, and we've heard about variant of unknown significance, meaning you'll have one additional way to classify those variants of unknown signification by having the allele resolution and the methylation status that tells you if it's expressed or not. So going now to germline and targeted, because whole genome means a lot of analysis, means a lot of data storage, but you can also do targeted. And there's a question I've asked to rare disease people. Is it better to go from a short read panel to short read whole genome, or from a short read panel to a long read panel? And actually, the answer to this one is not clear-cut, but in rare disease, they're seeing more and more that actually better do long reads rather than whole genome short read. And this publication that is from a cancer predisposition publication tends to prove that at least you get additional insights. It's a publication from last year. You see the reference on the top right. And what they were looking at were unresolved cases. Those families had been subjected to short-read panels, short-read exomes, and short-read whole genome. Then they did a long-read panel of only 10 genes, and they were able to show that in 88 of the 120 families, they were able to find deep intronic variants. And for 8 of those families, they were able to see that they could come to a conclusion as to what was the predisposition of those, why those families, why those individuals were developing cancer. So that's 6% diagnostic yield, or 6 points of percentage on top of what you classically get with short reads. And they're using a panel, but they are preparing a whole genome library. This is a technology that we call adaptive sampling, in case some of you have heard about it. It's a non-chip enrichment, meaning that you have a whole genome library, and then you instruct the sequencer to select, or the software to select, what fragment to sequence. And you do this by providing a bed file with your region of interest. And we just launched a panel that was inspired from work like the previous one, that is made of 258 genes that are genes known to be cancer predisposition genes. And because it's native DNA, you're going to get all the variants, not only the SNVs, but also the structural variants, copy number variations, and the methylation. You're going to have an average of 30x sequencing depth, but on the top of this, as you can see here on this diagram, you're also going to have a low-pass whole genome in the background, which means that you're going to be able to have a general information on, in particular, the methylation status and copy number variation across the whole genome. And you get this type of results classically, where you see here on at 60, so the y-axis is the coverage, and you see the region of interest that are well above 30x, but you see some are a bit lower, that's classic, and you see this low-pass whole genome there. And if you want to look, and one of the interests of this is to look at genes like that are difficult to map, either because they are from the dark genome or because they have pseudogenes, such as PMS2, that is notoriously complicated. And you see there results from the beta tests where they had PMS2 there. You see the 2 alleles, right? Here there's no variants detected. This is the pseudogene, and here this is the gene PMS2. And you see there that they observe a large deletion there, and you see also the allelic resolution, and you have also the methylation status, which would enable you to see if this mutation is expressed, or suggest if it's expressed or not expressed. So now, there is something that I want to say that is very different from what you know, and that needs to deconstruct a little bit what you know about NGS. If you receive a tumor, if you have a patient coming in and you have a biopsy, you're going to want to look at as many parameters as you want. And oftentimes, this requires different technologies. Here you have SNVs. You're going to do your NGS panel, structure variants. The NGS panel might be enough. You might need something more. Digital karyotype, and potentially methylation, as methylation is getting more and more accepted as a tumor classifier, meaning you can classify the tumor type that you get. It's for brain tumor in particular. So you're going to have those different aspects. And what ONT can do is, from one data set, getting access at all days, depending on the analysis that you're going to do on the data set, right? And not only this, but classically, when you have a cancer patient, you want to go as fast as possible to get your results. And one thing that I did not mention about Oxford Nanopore data is that you have access in real time to this data, meaning you don't need to stop the run to have your sequencing data. And there are publications that have shown that you can do in less than 1 hour of sequencing the methylation classification altogether from sample to this result. It takes about 2 hours usually. You can do a digital karyotype because it's a low pass copy number variation in, again, less than 1 hour of sequencing. But you can let the run go and get little by little more and more information as you go. And you use for this adaptive sampling approach that you've just seen. So prepare a whole genome library and boost the regions where you want to find your variants. And in early 2026, we're going to release this product that you can see that is for brain tumor classification in particular, looking at methylation, but also looking at some SNP and structure variants using an enrichment method in real time, the same one that we've talked about before. But as usual, we didn't invent anything. It's researchers who took ownership of the technology and ran away with it. And you have this great publication from a group of Dutch researchers working at Prinses Maxima Center in the Netherlands that showed how methylation and tumor classification using methylation makes a difference for them, right? Because they can do it in real time, including intraoperatively if need be. That is, the surgeon takes a biopsy, sends it to the lab, and they do the sequencing, and they can return the result as to what is the tumor methylation, the tumor type that the surgeon is operating. And you see there, they compared it with radiology, histology, and they see that their technology is non-inferior. Actually, they have less incorrect results, because either it's correct or mainly correct, or it's no result at all, which is, maybe better than giving a false positive or a false result. A second group from the UK has taken this technology and has gone further with this, which is what I was describing earlier. They look at the methylation status in 1.5 hours, but then they let the run go with some regions that are enriched, and they can look at structural variants and other mutations, translocations, etc. And recently this year, so you're going to see there are 4 publications, all from 2025, we've seen people from the oncology field taking this approach and looking at the methylation classification, doing digital karyotyping within 1 hour, and looking sometimes at more than just these. The different papers cover different aspects of this, because you can see it as a menu where you choose what you want to do. You can do everything, or you can do just one thing. So in conclusion, there's a lot of words, so I'm not going to go into this, but we can do a lot of, we can cover a lot of your needs in cancer research or cancer even diagnostic for very soon. I would say don't listen to what you hear about nanopore technology. A lot of people who tried it are very positively surprised, so just talk to us. So you can see at the back, guys, can you wave, please? You see those people, you need to talk to them. Yeah, you can wave as well. Talk to us, we're happy to answer to your questions. Thank you very much.
Okay, thank you very much. Any questions?
No? I'll let you close then.
Good. No, I was going to ask you something, but I can't forget. I've forgotten what yes. Your adaptive sampling panel with the 258 genes.
Yeah.
How much sequencing do you have to do for that?
So you're going to put 3 samples per flow cells, which means promethine flow cells. Yeah, so you need the larger sequencers, and you're going to get, so we think that you don't need to reload, so you need usually 15 gigabases per sample, knowing that 14 will be the low-pass whole genome and 1 gig of, yeah.
Okay, thanks a lot.
Thank you.
Okay, I'm delighted to see that some of you are still here, and I'm going to try to amuse you for the next, not that many minutes. I'll try to go quickly because I know you're all wrecked. You've gotten a lot of information plugged into you. So I gave this title to my presentation, Why Would You Go on a Journey Without a Map? So I'm actually only going to talk about maps today, nothing else. And to start off with maps, I'll show you this Beatus map that is from the Saint-Sever monastery. And this is very interesting. This is from 1050. This was drawn, and this was sort of copied and copied and copied. And if you look at this, so it's over 1,000 years old. No, it's 1,000 years old. it's not quite oriented the way we used to look at maps, but it's actually reasonably good. So if you look at the bottom, that says Occidents. If you look at the top, it says Orients. That's where the sun goes up, that's where the sun goes down, that's east, that's west. And you can obviously see that the thing in the middle is the Mediterranean, okay? Now, this map is extremely interesting because a map has things on it that you have an interest in, or might have an interest in. This one here has Eden on it. So on the top little square, you see Eden. So if you ever wanted to know where Eden is, that's where it is. You just have to take this map, you will find it. Now, obviously, you notice this is not completely correct. You know, it's sort of, you know, a little bit skewed and everything. But if you go to later maps, as this one here, which is the Catalan Abraham Kreiske's map, you see this is already quite, this looks quite reasonable for 1300. And the thing with this one is, this is like the Guide Michelin of the 14th century. This is actually only telling you where the good food is. So where the good places are where you should go buying your food. So this shows you all the things that are important. Now, maps can be constructed in very many different ways. Obviously, you can just take a measure and you can measure from one place to the next and then figure out, like, do the geography of that. But there are other reasons why you can have maps, or what you can do with maps. For example, this map is very interesting. Now, you're looking at the land and you're thinking about that. But it's not the interesting thing is the yellow things, the plains. What do these plains actually tell you? I'll tell you what they tell you. They tell you where the plains are. Okay, that's not particularly interesting. It tells you where the winds are blowing. So it tells you which is the best way to fly across the Atlantic to use a minimum amount of petrol going across there, or kerosene. So this is a dynamic map. This is a map that will change every day. It'll go up and down a little bit in function of where the winds are running. Maps can even be used to sort of identify political unrest. So you can see Venezuela in the middle there. There's no plains over Venezuela. Why is that? That is because the airspace over Venezuela was closed. And so you have no planes over Venezuela. So you can derive very interesting things from maps, and it's very important. Simon is fainting at this one. So, maps. Do you have to actually take a tape measure and measure all of the countries to figure out what continent you are in or not? Well, no, actually you don't. So this is probably for Simon, it probably is 2 weeks of work or something like that. For me, it was a rainy Sunday afternoon at home in the apartment in Paris. So Simon sends me sends me this principal components analysis. And what this is, he's taken whole genome genotyping data. And he's basically done a principal components analysis, and he's labeled the country of origin of the sample in a color and a symbol. So each color represents one country. I'll point out a few to you. The French are in red. So the French are red. Green on the bottom is, that is Norway. Okay, so if you look at this, so I noticed very quickly when I looked at this that if you just flip this around and projected it onto the map of Europe, you got the map of Europe basically by looking at the genetics of the people who live on this continent. So that is, you don't need to measure anything. You can just use the people who live here to tell you where you are. So this is good enough. So this is to me this is very interesting. And as we've gone through years and years of work, I actually always come back to maps. It's always maps. It's again a map of something. Good. So now I'll tell you a little bit of real science, but I try to keep it light. So about 7 years ago, we put a grant proposal to the European Commission for an ERC synergy grant. I did this together with Holger and Elias and Iñaki, and we basically said we were going to do the following. We're going to sort of do a single cell genomics analysis, comprehensive, of B-cell maturation and the transformation to chronic lymphocytic leukemia. So we wrote the proposal, we sent it, we were evaluated really well. We had to sort of refine something. sent it back again, were evaluated a second time really, really well. And then we were invited for interview to Brussels. So we went to Brussels. Oh no, we knew, so interview was just after the summer holidays, sometime in September, and this we got like in June, at the end of June. And we said, well, we would be far stronger if we had some pilot data. Okay, pilot. So we made pilot data over the summer. This is SmartSeq that we used, and this is 4 plates of sequencing, so it's 384 single cells analyzed. And what it is essentially one patient who goes from the initial diagnosis of CLL, he gets treated, he relapses, gets treated again, and relapses again to a third stage. Oh, there's blood, lymph node, blood, and then the lymph node at Richter transformation. Richter transformation is the form of CLL that then leads to your death. Okay, so we had single cells from all of those stages. So we did the single cell experiment, and what you can see is that essentially the samples coming from diagnosis, blood, lymph node, and blood after relapse, they cluster together quite nicely at the bottom. And the Richter cells, which are the ones on the top in Bordeaux, they are really well separated. So from an expression profile point of view, these things are different. However, if you look very well, you will see at the top right of that, you will see a light blue and a dark blue cell. So you see 2 cells, and those 2 cells, what that essentially is telling you is that the samples that were there at presentation of the disease that are like the Richter cells, they were already there years before the patient actually transformed and became a Richter transformed patient. So the thing that was going to kill him was there at the beginning, and basically because there was no way of figuring out that those 2 cells were there at the beginning, the treatments were not adapted to trying to clear out those 2 things. And when we went up to the committee and told them this, it was like, it was, les jeux sont faits. It's done. It was like, we didn't really have to say a lot more than look at that, and they said, okay, here you have the money. And so we took the money and we did an awful lot of work with that. And I'm really grateful to my colleagues, the 3 other PIs, plus all of the people who have been involved in this project for the work that's been done. So how did we start? So the first thing we did is we wanted to make a reference map. So a reference map meant we needed organs, in this case a secondary lymphoid organ, the tonsil, to sort of get a lot of immune cells. We wanted B cells essentially, but in the end we sort of widened this up to, well, let's just do all of the immune cells. If we're already at it, we'll just do everything. We'll take a lymphoid organ, take it apart, and we will do single cell analysis on that. So that was the first target. So to create a single cell driven taxonomy of cell types and states in the human tonsil. For this we recruited 10 donors who were happy to sort of part with their tonsils. As you can see, some of them were children, sort of adults, and then one fairly old person. Oh, this has the same problem in it that I wrote you an email about, Marianka. The hospital is a Swiss hospital. So you changed it. You changed it this morning in one of Marianka's slides. Okay, you see the little white cross on the red background. That's a Swiss flag. I'm Swiss, so when someone shows me that, I go, that's wrong, because actually it should be a red cross on a white background, the red cross. But this happens very often. Okay, it's an excerpt. No, if you look at it, it's about 40% of the time people use the Swiss flag for a hospital, and about 60% of the time they use the right way around. Okay, so what did we do? So we essentially got the single cells, and we did a multi-omic experiment on them. So we did single cell RNA sequencing, single cell attack, multi-ohm, which has 2 levels. It has RNA and attack in it at the same time. Site-seq, which measures the surface markers, the proteins that are on the surface of the cells, and we also did a spatial experiment. So we tallied all of that up. I think, yes. Now, just a quick excerpt into single-cell sequencing. So the first thing is, and this is probably to Roberto's experiment as well, one thing that you need to be aware when you're doing a single-cell experiment, it is a sparse experiment. So if you get a transcript that's coming from a gene that's expressed in an individual cell, it might be that you only have 1 or 2 molecules that are telling you something about that. And if you have 2 cells that are similar, they might not necessarily be expressing exactly the same transcript next to 2 cells next to each other. Might not be doing that, but they still might be similar because over their entire profile they are similar. So you don't get every gene nicely high expressed. You get a few reads that are sort of giving you like a glimpse on what this cell is actually doing. The next thing is that when you do single-cell RNA sequencing, for example, you are actually only sequencing into the end of the molecule. So, well, if it's done by paired ends, but it's sort of, you're only sequencing a very little bit. So from one end, which means anything that happens inside that transcript that is further away from the end, you are just not going to see. It's not there. And then that means, and this Christoph just said this, that actually you cannot identify the isoform that the transcript is coming from. You'd be very lucky to do that. So many genes sort of express in different isoforms, but you actually can't figure out which one it is. So in the experiment we did, essentially we had 3' and 5' sequencing, and so not complete coverage of the genes. However, the 10 donors, 5 modalities, we measured 350,000 genes. We got 137 clusters, and out of this, we got 121 cell types and cell states that we managed to identify. We annotated them. This will come again in a second. We managed to physically locate them within a tonsil, so we could figure out where they actually sit, where these cells are in relation to each other, and then we did a huge annotation effort. So this is, we first grouped it into 6 major classes of immune cells, and then manually annotated each one of these, and as I said, identified 121 cell types and cell states. So this now is the map that you can use, a very high resolution map that gives you an idea of what the cells of the immune system actually look like. I'm not saying that it's exhaustive, it's just where we stand today. This could be driven further, and you could go way further. But having said that, you also, you're just looking at, if you're looking at the transcript level, you're just looking at the end of the gene. So you're just saying this gene is expressed at this amount and finished. Which is when sort of Oxford Nanopore came to our help. So they came in and they said, we could actually do the analysis of your 10x libraries using the full length. So you're going to read through the full length of your RNA molecules. And so we took libraries, we hadn't fragmented them, that was very, very important. The DNA molecules, the DNA molecules that are there, they are full length, they are not fragmented, so they've been made in that way. And then they were made by reverse transcription. We've included another step, a PCR amplification step, to sort of get more of those reads in. We could do this gene specifically. I'll try to explain that to you in a second. And then we generated out of that an Oxford Nanopore library and sequenced it, one sample on an ONT promethine flow cell. So it's quite a lot of sequencing that we did for this, and then took all of this through analysis. So just to remind you, it's the same RNA-seq libraries that were produced here, but now we're doing them full length. So the first thing that came out, we made a map out of what we got, and this looked very, very convincing. Looked very, very much the same as it looked like in the short read, in the 3' tag sequencing. And so this is very promising, but obviously the thing we wanted to know is what does it look like in terms of usage of isoforms. And what we do see is we see many, many genes. There are at least 210 isoforms that we found that show huge spread, so differences splitting of the gene into multiple different isoforms that are used in different ones of the cell annotations that we have. Now we're at the moment still digging through all of this. It's an awful lot of data to do this, but I can just show you a few here. So see, this is one, a couple here, a couple here. One that is this is really neat is this PTPRC gene that has 5 isoforms, and what you can see is that you clearly have usage of one isoform in T cells, a completely different isoform in B cells, and then you have some intermediate ones that are sort of moving between one and the other. Okay, so now we have a map. So now we know what these immune cells look like. Now the next question is, so if we have someone who has CLL, can we actually figure out which cells are actually being used and whether there are differential transcripts on these individuals? Now, the question underlying this is, are we able to identify the cells that carry the driver mutations in a particular patient and grouping them into a sort of a pseudo-bulk RNA profile? Now, the problem with this is when you told you before that a single cell experiment is a sparse experiment. So that means you might only get 1 or 2 or 3 reads of that go across the full length of the gene. If you know that you have a mutation that the patient carries a mutation in a particular gene, you can now try to use that to look up that specific position where you know there is a mutation, and you can test whether the mutation is present or not in a transcript, and then use that to assign the cluster as a mutated cluster and then make the pseudo-bulk out of that. And that's what we did. So the first thing is, you will pick up cells, but very few cells are actually showing you the mutations because they are not necessarily expressing the gene that you are interested in looking at. So you know there is a mutation at a given position, but it's not necessarily expressing at that. But in the few that you get, you have to sort of do a fairly simple test. You basically do a Fisher test. You just say, I know there's a position here that should be mutated. Is it mutated, yes or no? And you just sort of attach a confidence score to that, and then you assign it to that. And what I'm showing you here are 2 different ways of doing it. In one case, we targeted the gene that we knew. So we knew there was a mutation in this XPO1 gene. We knew the position of the mutation. We did an amplification of the gene specifically for that gene and looked at that position. However, and then we did this with a MinION flow cell, so not that much sequencing, just to look at that targeted thing. The alternative is you don't bother about targeting, you just take everything, the whole transcripts, and then you do the same thing. And it turns out that essentially the numbers of cells that you identify as carrying the mutation, if you do the targeted approach or if you do the whole genome approach, is roughly the same, except that in one case it's a MinION flow cell, in the other case it's a PromethION flow cell, and you have to negotiate well with ONT to make those prices competitive with each other. Okay, so I'll show you some results. You need very good eyes for this, careful. So you basically take the long read transcriptomes of the individual cells. You make a UMAP out of that. You tell the UMAP to sort of give you clusters that have a certain degree of similarity, which is what you see on the left. So we just have clusters 1 through 11, and then you look for the places where the cells are falling that are carrying mutations. And it's a little bit difficult to see, but I'll show you this with a laser if I can figure out how to do that. Okay, so red is a cell that we are confident that the mutation is present in that cell. Blue is a cell that we cannot see the mutation, but we have coverage to read. That doesn't mean that it is not mutated, because humans are diploid, so you have 2 alleles, and usually only one of the alleles is mutated. Okay, but what you can see is in this zone here, there are red dots. So all the gray dots are all of the cells, so there's an awful lot where we don't have the information about the mutation state of the thing. But what you can see here is a group that essentially has red dots, which means you can now generate a transcriptional pseudobulk out of these two clusters, the yellow and the red, and then do sort of a differential expression analysis. So I'm not going to go into all the details of all of the results that we get out of this, but we're in the middle of doing this analysis. The point I want to make is you can detect somatic mutations. If you know where they are, it's easy, reasonably easy to detect them. If you don't know where they are, that's a little bit more tricky. So de novo somatic mutation detection from whole transcript full-length sequencing is challenging, but maybe that's doable at some point, maybe if you sort of generate more reads. Okay, and with that, I'll move on and show you a couple of pictures of the people who did the work. On the bottom is sort of the people with the tonsil atlas and the B-cell atlas project, and then Madeleine and Claudia who did the long read experiment that I just showed you. And with that, I'll sort of go to this particular slide here. In case you want to ask me anything, you're allowed to do that. Questions? That's good. No questions. Okay, let's move on. So we've reached the end of the day. I think it's been an extremely interesting day, very intense. I hope you haven't got as much of a headache as I have, but that's part of it. I want to say thank you all for coming and participating. I hope you enjoyed it, and essentially, if you want, you can have a look at our YouTube channels and all of our social media things that I don't even know what they are. And so, as I said very early on, we can only do this event due to the sponsorship of the companies Illumina, Nanopore, Agilent, Bonsai Labs, and Roche this year. So we appreciate this very much, and obviously our co-funders, which are the two governments, the one of Catalonia and the one of Spain.